just say some words in Swedish first, so to the student and to everyone who listens. So start with that. God morgon allihopa, ni som är här och ni som lyssnar på inspelningen sen. Jag vill bara påminna, ibland blir jag helt överrumplad av att jag inte har skrivit in planeringen. Det var helt tomt nu så att nu har jag snabbt skrivit in resten av den här kursen så att det finns på It's Learning. Och på måndag är det Lallemand på förmiddagen och på eftermiddagen. Sen är det självstudie på torsdagen. Den kan jag inlämningsuppgift. Och sen är det veckan därpå så är det också föreläsning av Lallemand. Och det är på måndag eftermiddag 2 stycken. Och det blev lite längre för det var enda sättet för oss att få in det. Så att det är 13 till 17. Så är det 2. Och det är jättebra föreläsare där. Så att det är kul att höra. Bland annat om skum och om vad det nu var mer. Så att det blir jättebra. Och sen kommer alltså kursen om destillering börja. Jag ska lägga ut planeringen för den också sen. Så det kommer också bli kul. So then I will welcome Andrew from Lallemand, and you can introduce yourself and so on. And thank you very much for doing this for us. It's a pleasure and a privilege. So.
No problem at all. Before I do, can I make sure that you can actually see the slides? Because I'm not sure which screen is sharing. You can. Great. So that's a good start. So we know what's going on. Excellent. So my name's Andrew. I work for Lallemand and have done for the last 6 years. So prior to that, I had a biochemistry degree and I worked first at Brewdog up in the northeast coast of Scotland. And then after that, I was up there for about 10 or 11 months. I moved to the southeast of England and worked for a reasonably large cask brewery called Darkstar, which was mostly doing cask ale. Although we did start to branch out into doing kegged beers and more modern styles whilst I was there. But we were probably doing, I think 400 casks a day, 5 days a week, for about 6 years. So it was, quite a fun process, and certainly taught me a lot about cask beer, which obviously isn't a huge thing in Sweden, but I have come across it in various places in Gothenburg. So I know it does get out and about. So after about 6 years at Darkstar, I decided to look for other things. And we didn't, at the time, really want to move. So I started to look at technical jobs. And I think some things came up with malt and with process aids, which looked all right. But I've always had an interest in yeast and fermentation. And when Lallemand came up, I sort of jumped at the chance. So I've been there now for 6 years and I enjoy it greatly. A company that's headquartered in Montreal in Canada. Were founded in the early 1900s, not supplying breweries at all. Started out as a bakery supply business. So they weren't actually making yeast either. They were buying goods and selling them into the baking industry. There's loads of products that you would use in baking that you wouldn't even consider, which I have learned something about since I started at Lallemand. But there's lots of things that bakeries need besides yeast. But later into the 1900s, they did actually start a yeast factory, and that's the original yeast factory that we still have in Montreal. It doesn't make any brewing yeast, it just makes baking yeast, and it's a very large plant. But as time went on, they started to branch out into other biotechnologies, and since then, it expanded greatly, and we have lots and lots of different business units supplying different industries around the world. We are primarily a biotechnology company, so we're selling yeast and developing yeast, bacteria, enzymes, etc. But the really big businesses aren't brewing at all. It's baking, winemaking, it's biofuels and distilled spirits, fuel ethanol production. And then if you look on the bacteria side of things, probiotics, so animal probiotics, food probiotics, things for making cheese, things for making salamis, anything that's fermented or requires microorganisms is pretty much what we do. Most recently, we've started to work in the realms of chocolate and coffee. So coffee cherries are fermented prior to being roasted. So we've got yeast specifically for coffee fermentations and also for chocolate fermentations as well. So it's really growing a lot. And they started to do quite a lot of stuff with plant care as well. So natural microorganisms that can outcompete things that might cause problems on plants and molds and things like that. So there's lots and lots and lots of things that we do. We are now a global company, so a lot of that growth has come from acquisition. So we have plants all the way around the world, but the most of the brewing strains are actually produced in Vienna in Austria. We've got a plant there which is specifically dedicated to brewing yeast production. And the reason the plant is there is because it's quite small. So brewing yeast, there's lots of different types of strains, and the actual volume the required for those strains is smaller than, say, baking industry, where they might require 20-ton batches. We require somewhere in the region of 2 to 5 ton batches. So it's quite a lot smaller as business goes. With regard to Lallemand as a whole, brewing makes up about 2% of what we do. So it's quite a small part of Lallemand, but we are a reasonably large team, so probably getting up towards 100 people these days. We've got representation in countries all over the world, myself covering Northern Europe, but then 2 in France, 1 in Spain, 1 in Italy, 1 in the Czech Republic, and then 2 in Australia, 2 in China, 1 in New Zealand, 5 or 6 in the States, where I'm thinking Argentina, Chile, yeah, Mexico, lots and lots of people. So it's grown a lot even in the 6 years that I've been with business. And it's quite interesting to see how it's changed and how we've started to do things slightly differently. And I've got slides coming up on that towards the end of the second presentation where I'll talk about what kind of things we're doing with regard to kind of development of new strains. Because that's been really interesting the last probably 3 years. We've started to do that quite differently. So today I'm going to talk about yeast and fermentation in general. So we'll start off with kind of a basic what is yeast, and then we'll look at types of yeast, how you might monitor fermentation, why yeast ferment in the first place, and then going on from that, we'll talk about how we make yeast, innovation in yeast strains, and we can move on and do some questions and stuff as well. So hopefully I won't be too boring, and you'll enjoy what I have to say. So without further ado, let's move on to the next slide. If it will let me change it, which it might. There we go. Did the slide change? It did. Perfect. Okay. So we're going to talk about what brewing yeast is. And you probably don't need to know this sort of thing in too much detail, but if you did high school biology, you might remember this sort of thing. So on the left-hand side of the slide, we have the 3 domains of life. We have the bacteria, the archaea, and then the eukaryotes on the right-hand side of the slide. And you can see right at the top of the eukaryotes, you have animals, which would be us, and then we have fungi, and yeast is actually a fungi. It's much closer genetically to ourselves than it is to a bacteria or even a plant. So we are quite closely related to yeast as far as all the life in the world goes. On the right-hand side of the slide here, you have the actual taxonomic classification of yeast. And you can try and pronounce all of those words if you really want to, but I think the things that are really important to remember are probably the genus and then the species. So the main genus used for brewing is Saccharomyces. Not always. There are some yeasts out there with different genuses. So one of the famous ones that sometimes used for low alcohol beer production would be Saccharomyces. But if we're talking about normal brewing practices, most of the time you're using Saccharomyces, and you'll either be using Saccharomyces cerevisiae, which is ale yeast, or you might be using Saccharomyces pastorianus, which is lager yeast. And we have a slide on lager yeast coming up where we talk about where lager came from, the genetic history of lager yeast, and how it differs from ale yeast as well. If you needed a mnemonic and you wanted to learn all these things, you have King Philip came over for great spaghetti. So if you're revising for any sort of exams and you need to know that sort of thing, that's something that you can say to remember the different taxonomic classes. So practically, what does yeast look like? On the left-hand side of the slide here, we have an electron micrograph showing yeast cells under the microscope, and you can see they're kind of almost spherical, spheroid. They're not quite round, but they're not quite ovals either. They have these distinctive markings on the outside of the cell, which are called budding scars, and they're formed when the yeast actually divide in half. Size-wise, They're about 5 to 10 microns, and you should be able to see them under a light microscope under 40 times magnification. And you'll be able to see some of the organelles that exist within the cell. So you can usually see the nucleus, and that's what makes a yeast different to a bacteria, among many things. It stores its DNA within a nucleus and not freely within the cytoplasm of the cell. They also have mitochondria, which are these little pink things, and they're involved in aerobic fermentation, so respiration, which is what we do. They'll have this big vacuole, which is a side of gas exchange within the cell. And then quite importantly, they have the cell membrane and the cell wall that surround the cells. So the cell wall provides rigidity or structural integrity to the cell. And the cell membrane acts as a site of kind of exchange between the outside environment and the inside environment of the cell. So anything that's floating around in the medium that the yeast happens to exist in this case wort, has got to cross that membrane to get into the cell. So membrane health in yeast is really key to their survival and also fermentation. So this is what a cell looks like if you zoom in on it. And there's lots of interesting things in there. And some of them can be quite important. All of them can be quite important, I suppose. So the main membrane itself is made up of something called a phospholipid bilayer, which if you look at the bottom here, the kind of gold circles with little tails. And the way that they work is that the circular heads are quite hydrophilic, so they're water loving and they'll face water. Whereas the tails are fatty acids and they're hydrophobic and they'll face away from it. They form a, if you did it just in water, it would form a micelle. So it form kind of like bubbles of essentially fat in a liquid. And it forms the main structure of the membrane. Within that layer, you have various things that will allow sugars in and out of the cell, or amino acids in and out of the cell, or salts in and out of the cell. And they're very important. But also within that membrane, you have things like sterol. And sterol is a component of the cell membrane that allows it to be fluid and allows that cell to be less rigid. So if you don't have enough sterol within your cell membrane, your cells can get rigid and they can start to break. So one of the things that yeast really require when they ferment is actually a bit of oxygen at the start of fermentation because it allows them to create sterols. So if those cells are dividing, they don't have enough oxygen, you'll end up with a sterol deficiency in your cell membrane and the cells will start to become less viable. Also important, particularly in the production of dry yeast, is this compound called trehalose, which is the little green spheres with the red surrounding on them. And that's actually a sugar, but it's produced as a stressor response in yeast. And it also helps to maintain the integrity of the cell membrane. So when you're making dried yeast, one of the reasons that there's less variety in dried yeast than there is in, say, liquid yeast strains is because when you dry them, you're putting them through quite a stressful process. In order to survive that process, they need to be good producers of trehalose. And that characteristic is quite genetic in its nature. So some strains are good at producing trehalose, some strains aren't so good at producing trehalose. And therefore, some strains can survive the drying process and others can't. So when we're talking specifically about dried yeast, which is what we produce, it's actually a very important component of that cell membrane. Beyond the cell membrane, you have the cell wall, and that's made up of primarily chitin and beta-glucan, but you also have associated nanoproteins on the outside of the cell, which are used for cell signaling. And Lallemand as a business actually make quite a lot of use of cell wall materials. So not so much in brewing, but we've recently produced a product made out of cell walls for brewing. But these cell walls can actually be broken down into different components and you can separate them and you can use them to make various ingredients for food. So a lot of things like flavor enhancers. So if you ever looked at vegan cheese, it'll probably say yeast extract on it. And some of that yeast extract will probably be coming from a cell wall component that has been separated out in one of our plants and then dried and can be used in that sort of application. If you ever go to Lidl, they do nutritional yeast flakes that you can put on salads and things, and that comes from one of our plants in Estonia. So they'll actually use an enzyme process to digest the cell and separate out into component parts and then dry them separately. Most recently, in brewing, we've produced a product which is enriched in nanoproteins, the idea being that you provide mouthfeel and also some haze. So it's kind of an alternative to, say, using maltodextrin or lactose as you might use in like a kind of hazy IPA type style. So it's something that we don't really or haven't really done in brewing, but we're starting to look at now more recently. It does exist quite a lot in wine already. So there's products for making, for instance, if you have like an acidic thin red wine, you can add yeast products to them, and it can add more fullness in the mouth. So you can make your thin acidic red wine taste a bit more full-bodied and almost like a slightly better quality red wine, if you know what I mean. So that's been done for many years, and works quite well. And we are now rolling that out into brewing as well. Oh, gone the wrong way. So we briefly touched on differences between yeast strains, and we talked about lager and ale yeast. Oh, I'm not sharing my screen, or I am sharing my screen. Yeah.
Yeah, you do, but
Yeah.
can I ask just a question, because we always
course.
want to hear about this, and it's about oxygenate the wort before pitching, and we say that when you use dry yeast, it's not as important as when you using fresh yeast. Are you in the same opinion, or?
Yes, absolutely. And the reason is, if we go back to the previous slide, when you propagate yeast in a brewery, it will tend to be an anaerobic process. And yeast, when they're being grown, require oxygen to produce sterol. So when you're producing dried yeast, what we do is we try to keep it in a kind of semi-aerobic sort of nature of the fermentation. And we'll go into why that's the case later. But because that is the case, you'll find that during the propagation process for dried yeast, the cells are actually enriched in sterol to a greater degree than they would be in a kind of a brewery environment. So the level of sterol is such that they don't necessarily need oxygen to start fermentation. For the first generation, if you then repitch them, then they do require oxygen, because that sterol will be depleted during the fermentation process. And it isn't so much depleted, it's when your cells divide, your membrane is then essentially diluted in half, right? So the sterols, each division, the sterol level is diluted when the cells divide.
Great. It's always good to hear so many words about this many times. So
There's no massive downside to oxygenating either. So it's it's one of those things. If you wanted to do it, you can. You might end up with slightly too much yeast growth, which can cause some issues, although only really in a constant repitching process. Or if you pitch too little yeast, it can help quite a lot. So if you haven't used it enough, then you can use a bit of oxygen, and that will help. So one of the kind of fermentation problems I do come across, especially on second generations, if you don't oxygenate enough, the main symptom of that is that you'll find out that your fermentation is stunted, so it doesn't complete fermentation. That's because the cells have started to become not very vital, so they might still be alive, but they're not actually fermenting efficiently, and you'll find that your fermentation doesn't complete as you would expect. Moving on to lager yeast. What is lager yeast? Saccharomyces pastorianus is actually formed from a hybridization event. So you can think of this essentially as breeding two yeast cells together, much in the same way as you might breed animals together to create a new type of animal. In this case, we're actually breeding two different species together. So it'd be like when a tiger and a lion breed together and they create, like a liger or a tigon. So it'd be a bit like that. Okay, so it's a hybridization process that has taken place naturally in the environment, and possibly took place twice. We're not quite sure, but this has been known for a long time. So it's been known for a long time that the strain of lager yeast is actually a hybrid. What hadn't been known for a long time was the parents within that particular hybrid. So one of them was ale yeast, Saccharomyces cerevisiae. That was known. What wasn't known was the other parent, and that wasn't discovered until quite recently. I think that was 2011. A paper was released where they discovered this parent living in the wild, I think in Patagonia initially. So not anywhere near where the hybridization event is thought to have occurred, because lager yeast obviously is famous for the lager production, which is historically linked to Germany. So you expect that hybridization event to probably have taken place somewhere in a brewery cellar in Germany. But the first place they found this parent strain was actually in Patagonia in Argentina. But since then, they have found it living all over the world. So I think in Ireland and Tibet and Wisconsin, not yet in Germany, but that's probably just a case of not looking hard enough, I think. I think at some stage it probably will be found there and possibly everywhere as well. But there are two distinct groups of lager yeast, which probably came from either two separate hybridization events or concurrent events. So you had one that took place first and then another one that took place in that strain later on. And the two broad groups known as Group 2 or Group 1, or Froberg or SARS, and sometimes known as 3470. We don't like to call it that because one of our competitors has a strain called 3470, but it is correct to call it that. So you've got Group 1 and Group 2. Now, if you've ever brewed with lager yeast, chances are you've used Group 2. Group 1 is the original Carlsberg strain, but it isn't used very much in fermentation. Interestingly, the original parent, Saccharomyces eubayanus, Heineken actually brewed a beer with it for fun a few years ago, which is the one on the bottom right of that slide there. So that's called H41. And that was brewed with Saccharomyces eubayanus. I think, can't actually see what's on my next slide, but I think my next slide goes into some justification as to why that's quite tricky. Well, actually, no, it doesn't. So I'll go into it in a bit more detail. So the parent, Saccharomyces eubayanus, was a wild strain and was quite cold tolerant. So it liked the cold a lot. It was probably POF positive, so it created a phenolic flavor as it fermented, much like Belgian strains do. And because it was wild, it likely didn't ferment all the wort sugars, because fermenting wort sugars is something that's only really appeared in brewing yeast relatively recently in genetic history, because those sugars don't really exist in the environment. So there's no reason for a brewing yeast to ferment them. So if you're a wild yeast fermenting maltose or maltotriose, which are the main sugars in a brewery wort, is mostly pointless. It's not very genetically useful. So you'll probably ferment something like fructose or glucose efficiently, but not maltose and not maltotriose. So in the case of Saccharomyces eubayanus, if Heineken were to brew a beer using this particular strain, they probably have to do quite a lot of fiddling around with the wort to make it so it was one, fermentable, and two, tasted quite nice. So it would have been a very interesting project to be involved with, actually. I think would have been quite fun, but I don't believe they make it anymore. So I think it was a one-off project for fun. So more broadly, what are the practical differences that you need to know about between lager yeasts? So lager yeasts are actually less flocculent in general. That's just a genetic characteristic of lager yeasts, but they do flocculate, just not as efficiently as ale strains do. And traditionally, they would tend to sink to the bottom of the tank, so they ferment near the bottom, whereas a traditional ale would rise to the top. With the advent of cylindrical tanks and pitching from the bottom of those tanks, that distinction doesn't really exist too much anymore. So most ale strains would also ferment and drop to the bottom of the tank and be repitched from there as well. Probably the distinction there is some traditional English ale breweries and Belgian ale breweries where they'll skim from the top of the vessel and then repitch in that way. And they'll usually use either, if you're on the small scale, like you could use a spade with holes in it to pick the yeast off the top and then put it in a bucket. At the larger scale, they use suction lancers and they'll suck into a tank. So both strains can actually grow at low temperatures generally. It's just that lager yeasts are much more efficient at growing at cold temperatures than ale strains are. The real difference between them is the maximum growth temperature. So ale strains have a much higher maximum growth temperature than lager strains. So one of the ways that you can differentiate between the two in a lab would be to look at the maximum growth temperature, not the minimum growth temperature. And we have ale strains that will ferment at 10, albeit slowly, but our Nottingham strain, which is a bit of a workhorse of yeast, can grow at 10 degrees, and it can grow at 20 degrees quite happily as well. So it's actually quite flexible as yeasts go. You would also need to pitch it at a greater rate at cold temperatures because it's going to be metabolically slower. Anyway, so, Alexa, turn on the lights. You're getting put into the dark. Lager produces more sulfur compounds than ale strains. So as you ferment, you'll recognize if you're near a lager fermentation because you get that eggy characteristic being produced. Lagers tend to produce that at a much greater quantity than ale strains, but also because they're fermenting colder, it's also more soluble in the wort or the beer than it would be in an ale strain, so it doesn't flash off as easily. So there's two reasons why you get higher levels of H2S, that eggy flavour in lagers. As far as diversity goes, genetic diversity in lager strains is actually not very great. And one of the reasons for that is because of Christian Hansen, the famous scientist who worked at Carlsberg, when he invented the pure culture machine. And when he did that, he purified lager yeast and he sent it to breweries all over the world, Europe and the Americas and the New World as well. And because that was the case, a lot of breweries are using lager strains that are actually genetically very similar. They might be slightly different, but there's much greater diversity in ale strains than there is in lager strains because of that process. One of the places that were a real stick in the mud was actually the UK. So there's a lot of breweries in the UK that refused to use the pure culture mechanism created by Christian Hansen because they were a bit backward. But nowadays it's the norm to use pure culture yeasts and not tend to have a mixed culture, though some will. So some traditional Belgian breweries and quite a lot of the traditional English cask ale breweries are still on mixed cultures. So how does yeast divide? I do have well, I've got some notes on this one. Let's see if I can find my notes because they're on a different page. Just because I want to be able to talk about. There we go. I can't see your faces for a second, but I'll bring them back at some point. So they actually divide by a process called multilateral budding. So that differs to some other yeasts that might reproduce by binary fission. So that actually split in half. These kind of grow into kind of a separate daughter cell, and then that buds off the cell. If you ever look at yeast under a microscope during fermentation, you can see that process taking place. Each one will take place about 3 to 5 times, and it will create mother cells and daughter cells. Now, those divisions can take place up to 20 times. But as that cell divides and gets older and older, it starts to become less vital and eventually will die. So they don't last forever. And that can be important in fermentation. It's not something that you necessarily see very often, but if you were to be repitching yeast and you were to overpitch over and over again, because the level of yeast in that fermentation is so great, you won't get as many cell divisions and you'll get an aging cell population, and therefore you might find that your fermentation starts to become less efficient over time because you're pitching too many yeasts into that fermentation. So overpitching can be a problem over the course of several generations. If you did it once, probably no big deal, but if you did it 5, 6 times, you might find that you start to have a problem. So it is actually important to pitch at the correct level, not overpitch, not underpitch, but correct level of pitching, so you actually get some cell division and you get a refreshing of the cell population. And if you do that and you do it well, you can actually maintain for many generations. Particularly in the case of top cropping, because you're top cropping, you're actually pitching from the least stressful part of the fermentation tank, and you can get yeast to last for hundreds of years, albeit you do have to take some other precautions as well. So it's quite an interesting process. The left-hand side of the slide, you've actually got what happens genetically. So you've got basically DNA replication that takes place within the cells, a process called mitosis, and then the DNA will move to opposite ends of the cell, and then the cell will then split apart and it will form 2 daughters. So if you were doing your high school biology, you would need to know all those different phases of cell replication. And I think you probably agree.
I want to ask.
Go on,
Yeah, or just to clarify, because it's very interesting, because we especially work with microbreweries and when repitching we using from cone to cone, a
Yes.
volume, and I can imagine that's a risk that we overpitch when we take from cone to cone. Is that what you? So if we then do that a lot, then the fermentation slow downs and so on.
That's right. And it will work. What you probably limit is your the number of repitchings that you can do. So you can probably do that for 6 times, 7 times, no problem. But if you try to go 20 times, it's going to be more of an issue. Yeah. So it's people always ask you what's the downside with overpitching, and the truth is, if you did it once, there isn't really much of a downside. But if you did it a lot of times, it becomes more of a problem. It becomes more problematic.
Oh, that's great.
So I'm going to do a little bit on kind of types of yeast as broad categories, because I think this is one of the ways that you could classify your strains into kind of broad groups. And there are people that have done a lot of work on this genetically. So there's a university in Helsinki called VTT, and they've actually looked at the genetic differences between these large subgroups, how close they are. And it's fascinating. I'm not going to go into that level of detail here. I'm just going to talk about them as broad subgroups. So first ones I'll talk about are Weiss yeast, so strains from southern Bavaria for making wheat beer. This is kind of a traditional wheat beer fermentation vessel on the right hand side of the slide. So you can see they've usually got open tops. And there is a reason for that. It's because with an open top fermentation, you don't really get much in the way of hydrostatic pressure, and you actually favour ester production. And that's one of the things that wheat beers are very famous for, is ester production. So they produce an awful lot of acetate esters as they ferment. So you get like kind of banana, you get solventy, bubble gum, kind of hair drops from the fermentation. And you will also get phenols as well. But in a traditional wheat beer, you're probably more looking at the esters than you are the phenols. So you get really kind of fruity characteristics from them. And why is that the case? It's genetic. So all strains differ in their ability to produce esters. So I've put up here, this is the flavour wheel that we produce for our Munich Classic wheat beer yeast, which is the Klosterberry Andex strain. Have you ever heard of that brewery? That's a monastery brewery just outside of Munich. But some of the strains that we produce, we license from the Doemens Academy in Munich. So it's their strain banked with Doemens, and it's very, very banana forward. It creates loads and loads of esters as it ferments. And it's because it's genetically, it's programmed to produce lots of esters. So they produce enzymes called AATases, or I think it's acetoaminotransferases, something like that. Was it alcohol? I'm trying to remember the name of the actual enzyme, and I've forgotten, but it's AATases is the acronym, which is probably what you need. But if your strain expresses those particular enzymes at a high level, which wheat beer strains do, you'll naturally get lots more ester production during fermentation. So if you wanted to produce a wheat beer strain, you've got to choose the right strain. There are things you can do beyond strain choice to favour ester production, but the real thing that you need to do is produce or use a genetic wheat beer strain for that fermentation. They tend to be very vigorous strains as well, so they produce a huge throppy head during fermentation. They top crop better than they bottom crop, actually, because they do produce that big head. So if you had a traditional open top tank, it'd be quite fun to witness, because you could see it foaming everywhere. And you'd have to make sure that you didn't fill the tank too full, because otherwise you'd have it spilling down the sidearm of the tank, and you'd lose quite a lot in the way of volume. So quite similar to wheat beer yeasts are Belgian ale yeasts. So quite similar to wheat beer strains, but probably produce slightly less in the way of esters. That's not the case across the board. So some of them do produce quite a lot of ester flavours. But the flavour that they do produce, and are quite famous for producing, it's called 4VG, and it's a phenol flavour. And classically in brewing, you would call it phenolic off flavour, or POF. So strains that are POF positive produce this phenol characteristic when they ferment. And that's also actually a sign of wild yeast infection. Wild yeast tend to produce phenols as well. And the reason it's a sign of wild yeast infection is actually because that characteristic is very, very common in wild yeast. So over the kind of the centuries, as brewing yeast has evolved to become more domesticated, that characteristic has been lost from most strains. And that's actually just because people don't tend to like it. So people would tend to favour beers produced that didn't have the phenolic off flavour, compared with strains that produce beer without that flavour. So most of the time people don't want it. It is, of course, completely acceptable in Belgian strains or wheat beers, or German wheat beers. But in, for instance, like a lager or an English ale, it would not be quite so acceptable. If you have that flavour and it's more medicinal than it is clovey, then it's more likely to be from wild yeast than it is from like a Belgian strain. And the reason for that, next slide, this is the chemical that is created. But it's formed from chemicals that exist within barley and malt. So ferulic acid, and some of you will probably have heard of the acid rest, which is often done in wheat beer. That is a mash step. So what you're doing in that mash step is actually you're releasing free ferulic acid from plant cell walls. So that chemical actually exists within plant cell walls as a defense mechanism. So it protects those cell walls or those cells from attack by microorganisms. And if you mash at 40-45, there's an enzyme called ferulic acid decarboxylase that actually releases free ferulic acid from those plant cells, and then it is free within your wort. And it being free within your wort doesn't mean that you'll get 4-VG. So in every wort you'll have free ferulic acid. It's just that some yeasts are able to convert that free ferulic acid into 4-VG, which is the phenolic off flavor, and those are Belgian and wheat beer strains. So if you fermented with an ale strain, you wouldn't get this flavor, even though the ferulic acid exists. Likely there will be some 4G there. It's just that it'll be below flavor threshold, so it exists within the kind of overall chemical makeup of your beer, but it does not take part in the actual flavor because it's there at too low a level. If you boiled for a very long time, you might actually start to get it, because it does actually thermally break down into 4-VG as well. So if you boiled ferulic acid for long enough, you do get some 4-VG production, but you'd probably have to work at getting it at such a high level that you could taste it. So this is what it looks like. Ferulic acid goes to 4-VG and CO2. So this is what those strains are doing. So that's called a vinyl phenol, 4-VG. If you were to take another starting acid, and this takes place a lot in wild yeast, there's another starting acid called succinic acid, rather than ferulic acid, and that's the one that produces the kind of medicinal flavor. So it produces a different chemical, which is called 4-VP, 4-vinyl phenol, which is more kind of medicinal and dentist cherry. It's not a very nice flavor, and if you ever do get a really big wild yeast infection, that's quite often the flavor that appears first. So I mean, from personal experience, when I worked at Darkstar, I had a 80 hectoliter fermentation that somebody forgot to pitch. And you're monitoring fermentation by gravity. You don't necessarily notice on day one. Day two, it might be too late, and in this case it was, and we had a wild yeast infection. And while it fermented fine, the flavor profile was such that you had quite a high level of 4-VP, and you couldn't send it to trade. It was awful. So it went down the drain. We'll come to Brettanomyces yeast later, but that 4-VG can actually be further broken down by yeast, and it produces the flavor that's sometimes known as horse blanket. It's 4-EP or 4-EG, can be produced by Bretta strains to produce that kind of barnyard type flavor. So slightly more boring. English and American.
I want to question again. Sorry, and interrupting you so much, but
no, it's fine.
This is not actually about clove, but we met on this last Monday, and we tasted beer, and one of the students, Theo, he had a beer, and I think it's a phenol because it was so smoky, and it tasted like a smoked sausage. Is that a phenol? Can you imagine why it's that? Because he didn't had any smoked barley as a malt or something.
That can be a phenol type flavor, yeah. So if you're having smoked Scotch whiskey or peated whiskies, that's a phenol that produces that flavor. So it can appear quite smoky. I would suspect if it was in a small brewery and it wasn't coming from the yeast strain, it's probably burning. That's quite common. So if you've got electric elements and if you're using steam, the temperature difference between your element and the surrounding work is actually reasonably low. Steam's probably 110, 120 degrees. Electric elements get way hotter than that. So if you have an electric element, you actually get wort singeing. That's the most common source of kind of a smoky burning flavor that I get in beer from smaller breweries, and it's one of those that it isn't too intrusive until you notice it, and then you're like, oh, and it's quite noticeable. So the real key thing is make sure the elements aren't too hot, but also make sure that they're clean. So if they've got any burnt wort on them at all, you can get that sort of scorched, smoky burnt flavor from them. Obviously, the other one is smoked malts as well. So using peach smoked malts from Germany or peat smoked malts from Scotland as well, they produce that sort of flavor.
Yeah, thanks.
Right. So English and American ale yeasts, they're slightly more boring than German and Belgian yeast, I suppose, but they're actually a very broad group. So they can be very clean. They don't produce very much flavour at all. For instance, our Nottingham strain ferments at quite a wide range of temperatures, but doesn't produce very many esters at all. they can be really ester forward, so they can be really fruity. So a lot of the strains that are used for, let's say, hazy IPA production, things that spoken about as the kind of New England yeasts, they have a genetic history in the United Kingdom, and they can be very, very ester forward. So some of those cask ales are quite ester forward. They can be quite interesting, but some can also be very clean as well. They tend to be quite high attenuating, but not always. So you can get strains that don't ferment malted trios, and therefore they'll do a different range of attenuation than other strains that might do up to 75, 80%. Some of them might do 65, 70%. That's because they're not fermenting malted trios. And again, that would be that'd be almost like a lazy strain, so like a lazy cask ale strain. You quite often find with them that there'd be a mixed culture, and they'll have one strain that doesn't necessarily ferment all of the sugars and one that does, and they might end up pumping it over. So that's not something that you do very much in modern times, but you'd actually pump the work around the fermentation dust and actually get some oxygen into it to keep it going, because the strain could be quite lazy. Yeah, they're a very, very broad group, but I think one thing that does kind of probably unite them all is they're exclusively off negative, so they don't produce any phenolic off flavor at all. So there's lots and lots of diversity, but I can't think of any that produce phenolic off flavor. So that's probably the main difference between them. The two examples I've used are BOI 97, which is we describe it as a West Coast American ale strain, but it's very clean fermenting, doesn't produce much in the way of ester character at all. You can kind of think of it as a blank kind of canvas for hopping, really. So that was picked up by breweries on the actually the east coast of the United States, and then brewed with on the west coast of the United States. So it's kind of like your Chico, Sierra Nevada type yeast. And it actually comes from the Siebel Culture Collection. So Siebel is a brewing school in Chicago, which Lallemand actually owns. And some of our more traditional strains come from their strain collection. Windsor is a more traditional English ale strain, so that produces quite a high level of esters. So I'd probably relate it to a New England ale strain. Where it differs is that strain doesn't ferment malted trios, so it's got much lower attenuation range. And for that reason, some people will use it for alcohol or low alcohol beers, because it can naturally produce a lower level of alcohol. That strain's got quite an interesting history within Lallemand in that it was produced for Boots the Chemist, which is a chain of chemist shops in the United Kingdom, which used to make the homebrew kits. They don't anymore. So they approached Lallemand in the mid-90s, and they used to always sell their kits with fresh yeast, with liquid yeast. And they said, would you be able to dry our strains for us? And I think Windsor was the first strain that we dried for Boots the Chemist. So that's the kind of background behind it. Going beyond the English strains, we've got diastatic yeasts. So this would kind of be probably like a subdivision of Belgian strains, I suppose. So it's used to create saison, which would be in that kind of northeast France, Belgian border kind of region, which is famous for saison. And they're quite interesting strains because they are diastatic, which has been quite a big subject in brewing over the last probably, I guess, 10 years now, really. But they're unique in that they can ferment dextrins. So if you look at this slide here, you've got on the left-hand slide the main sugars within brewing fermentation. So glucose, maltose, and maltotriose. Most brewing strains can ferment all of those. Most brewing strains can't ferment anything longer than maltotriose. Anything that's a dextrin, anything that would, I guess, make up the residual gravity at the end of a fermentation, can't generally be fermented by brewing yeast, but it can be fermented by saison strains or strains that have this diastatic phenotype. So they have a gene called SDA1 that produces an enzyme which will actually break down those sugars and create fermentable sugars, which the yeast can then metabolize and create further fermentation. So very good if you're trying to make a very dry beer in your brewery, or you're doing mixed fermentations. Not so good if you're creating unpasteurized beer and you don't want there to be further fermentation, let's say in your packages or your kegs, because these strains can continue doing this for quite a long time. So they can break down these dextrins over a period, and you'll find that your cans start to become overcarbonated, and in extreme circumstances, they can actually produce so much CO2 that they pop. Which obviously is something that we want to avoid. So In recent years, people have started to use them less, particularly when people started to have their own canning lines in-house, because you really don't want that to be contaminating your other products. So it's an interesting one. We've produced a new product, which I'll go into later, where we've actually taken a saison yeast and we've bred out the ability to ferment dextrins, so you can create saison-like flavor without the risk associated with it. However, if you're very, very safe, it can still be done. I personally wouldn't put it through my own packaging lines unless I was pasteurizing afterwards. But people still do. On one note on that, I'd probably say is that if you measure the gravity before you package and you're packaging unfiltered beer, if you then measure the gravity in that packaged beer after like a month, you'll probably see that it's dropped by 1 degree anyway. And that's usually residual maltotriose that's being fermented. So all strains have a differing ability to ferment maltotriose, and you might find that there's a little bit left. So it wouldn't be completely unusual to have a little bit of fermentation in your can, but to have a lot of fermentation in your can is a problem, like a big problem. Be careful with diastatic yeasts. This is something that we do or are doing constantly a lot of work on within our plant. So we produce one strain, Belle Saison, which is our only diastatic yeast. Sales of that over the last 10 years have collapsed, I would add. But we actually produce it in a different plant. So most of our brewing yeasts are produced in Vienna in Austria, and the Belle Saison is produced in Passau in Germany, so it can't contaminate the brewing yeast. And we've also actually discontinued it in 11 gram for home brewers. And the reason we did that is because we only have one 11-gram packaging machine, and we didn't want to put that anywhere near it. So it doesn't exist in homebrew format anymore because of that reason. So it's something that's really like come to the fore over the last few years. People have started to become really concerned about it, and rightly so. So it's something we spend a lot of time searching for, probably the most time out of anything searching for that.
And just to be clear, because I got a question from a student before, and this is a regular. Let's say Windsor yeast. They can't mutate and start to be diastic. It's specific for some yeast strains. Yeah, because we know all this talking about in the United States when breweries started to get diastatic cells and so on. It's not mutation; it's just then it's contamination. That's
yes, yeah. I mean, it's an inherent kind of characteristic of the strain that it would be able to do that. I mean, to add further complexity to it, one of the ways that you'll look for this is using PCR, or polymerase chain reaction, to look for the actual gene, because that's the way to find it. But there's been some research recently on these strains where they actually have the gene, but it's not active. So it becomes slightly more complicated or trickier the closer you look at it. But no, for the most part, there will be a characteristic that the strain has anyway, not a mutation of, say, the parent strain. So if it's there, it's probably a diastatic yeast that's got in somewhere. And they're hard to detect. They grow and look very much like brewing yeast, so it's very hard to differentiate from those brewing yeast. The only real way to do it is genetically. We have lots of genetic methods, and we've done lots of kind of more microbiology based methods over the years to look at it, and it's really hard because one of the things you're looking for is probably excess gas buildup, and you might put that cell in like a starved environment, but actually some cells can still produce CO2 from intracellular carbohydrates, so you might still get gas production if you're looking in that way. So it's tricky. I think the Brewers Association in the States is trying to come up with like a unified standard at the moment, but haven't reported on it yet. So there's people always looking at it.
And then a follow-up question, because we talked about with the White Labs also, and they have started to make dry yeast just a little bit, and they said that dry yeast can be more what's the word? Please, none.
Contaminated.
Yes. Yeah.
Than fresh yeast. Is that your opinion also?
Yeah, it can be. Yeah, I mean, part of that is the drying process. So, because if you think about dried yeast, if you put it into a drying plant and then you try to clean that drying plant with liquids, it would turn to glue. So the way in which you clean the plant is quite different, and it's not 100%. So most likely is contamination with other brewing yeasts, which is why we produce the Balsaison somewhere else. Contamination with bacteria, not so much of a big deal. That's not such a big deal. But you've also got quite a big fermentation. So I think most of the time the liquid yeast are produced on a very small scale. We're talking more like, 20,000 liters is the small tank. So the opportunity for contamination is greater. So you'd never really be able to get zero, if you know what I mean, because it's kind of, I guess, fairly impossible at that scale to get zero, and we just have to get it as low as we possibly can. And we have very high specifications, but it's never going to be completely zero. So yeah, it is true. Yeah, it's definitely true.
Yeah, I think it's good to know because if you repitch a lot with dry yeast, then of course if you I can't say that word, but yeah,
that's the case, and we do a very long QC process, so we're probably about 2 months in QC, which means we can guarantee that our yeasts are able to be repitched. Not everybody does that, so not all dry yeast is created equal. So it's all about the QC process. Making yeast doesn't actually take very long. It takes probably 3 or 4 days. It's the QC process that takes ages. So we can have a batch produced in no time, but then actually releasing that is a very, very long process. And we do work both in Vienna at the plant, and then most of the samples are shipped off to Montreal, which is where most of our labs are, the QC labs are. So they do an awful lot of work there. And I do have a slide in it, but it's not just diastaticus. There's wild yeast, bacteria, fermentation performance, color, like all sorts of different things that they look at. So final broad subgroup, it's Brettanomyces yeasts. So not often used as a primary strain for fermentation, although you can. Some people do in the States. I probably wouldn't myself. But they can produce quite interesting esters. They can produce quite interesting characteristics. And part of that is the inherent ability of the strain to produce esters. But part of that is also the fact that they tend to be used in spontaneous fermentations. And in spontaneous fermentations, you get esters formed from lactic acid, which can be quite nice flavors, so kind of rose, strawberry, and that sort of thing. So they can produce quite interesting kind of fruity characteristics that you don't necessarily get from other brewing yeasts. One of the things that's quite misunderstood about Brett strains is that they don't produce a lot of acid. They'll produce some acid, but they're not producing the level of acid that you might get from, let's say, a lactobacillus strain or a pediococcus strain. They produce loads of lactic acid. This will produce some organic acids, maybe a bit of acetic, maybe a bit of lactic, but not loads. They're quite slow growing, so they won't necessarily be obvious in fermentation at the start. So one of the reasons these was discovered is because in Casco and Britain you'd find that the fermentation slowly continued down and down and down, and the causative agent would have been Brettanomyces yeasts. And they can also ferment a vast array of sugars. It does depend on the strains. So there's loads of them, like with ale strains, loads of Brett strains, but some will ferment cellulose, and some will ferment galactose, and some will ferment starches. So they really depends, but they can often ferment a lot of different things. Not really used much outside of brewing. Saccharomyces yeast are used for hundreds of different applications. Brettanomyces yeast, not really. Mostly just brewing and mostly just spontaneous fermentation beers. So beers that you would get in Belgium. So probably not a big thing amongst the craft brewers either. I mean, I think it's a market that used to exist, and people maybe overestimated the size of that market. And we've seen quite a few of those spontaneous-based breweries closing. So Brewdog had a really big wild fermentation program, as did Baghaven and Copenhagen, and both of those have since closed. And I think the reason is that there's just not that much market out there. If you think of Cantillon, it's not a big brewery. It's a very small brewery. And there just isn't that much demand for that style of beer, particularly in Europe. I think in the States it's actually different. They seem to be able to support that market better, but maybe it's just a case there's more people in the United States. It could be that there's more people to share it out with. So it's not used a lot. It tends not to be produced in dried form. We don't produce it in dried form. One of our competitors does, but we don't want it anywhere near our plants because of its ability to break down various sugars. So it would be akin to producing Balsaison in our brewing yeast plant. We don't want to do that. So The only place I think we could potentially do it would be, we have a plant in Brazil that makes some of our GMO strains for the States, so we could possibly do it there. But really, we don't think the demand is there for it, so it's not something that we would produce. So if you do need it, I would probably get it in liquid form from White Labs. That's probably what I would do. Okay, so a bit on fermentation-based flavors and monitoring. So very little bit on the biochemistry of yeast growth, because yeast. Oh, go on.
We'll take now a 10 minutes break. I think it's
10 minutes. Yeah. Okay.
Yeah. So 10:05.
10:05. Perfect.
Okay with you. So
9:05. Yeah.
Here it's 10. Yeah.
Yeah.
Okay, so we take a break.
Great. I'll get a coffee.
Coffee. So, please continue.
Yes, I hope everybody got coffee. So you'll need it for this bit. The biochemistry of yeast growth. No, I'm not going to go into a great deal of detail, but one of the things that's quite interesting about yeast is that they're able to both respire, so breathe like you and I, or ferment. And when organisms do that, they create a chemical, which is the kind of unit of energy transfer within biological organisms, which is called ATP. And also on the right-hand side of the slide here, you can see the amount of ATP produced during fermentation and the amount of ATP produced during respiration. And you can see that one is far more efficient than the other. And the question with brewing yeast is, why did they do this? Why did they ferment when they could respire? Because yeast, even in the presence of large amounts of sugar, or in the presence of large amounts of sugar and oxygen, will tend to ferment rather than respire. So they deliberately do something that's inefficient rather than something that is more efficient. And it's called the Crabtree effect. It's something that we need to be very mindful of within our yeast plants, because when you're trying to grow yeast, you have to force them to do the thing that's efficient and not the thing that's inefficient. So the reason they do it is because there's a byproduct of fermentation called ethanol, which is the reason we all quite like to drink beer and wine and spirits. But for a yeast, it's it's useful because it acts as a competition factor with other organisms in the environment. So essentially, what it is it's a poison, and they're killing off other microflora around them in that environment by producing the ethanol, which is thought to be why they will tend to ferment rather than respire. So the whole reason that they're creating this ethanol, it's not for you or I, it's to kill off other things around them and steal all the sugar for themselves. Obviously during fermentation, you get ethanol production. So you get sugars, which are converted to ethanol and to carbon dioxide, which are the bubbles in the beer, but also lots and lots of flavor compounds. So we'll talk a little bit about some of the important ones now, because they're quite useful. This is kind of how it looks like in a cell. This is way more detail than you need to know. But on the left-hand side of the slide there, you have the classes of chemical that produce most of the flavors that you might taste in a beer. So the first one, higher alcohols. Those are the kind of hot flavors that you might get. They tend to be associated with warm fermentations or very high alcohol fermentations. Both higher alcohols and esters tend to be found in greater amounts at high gravity. So if you're producing a very high alcohol beer, or you're producing a very warm fermentation, there's lots of yeast growth, lots of these chemicals are produced, and you can't prevent them being produced. You can have a go at controlling them, but they are inherently associated with yeast growth, and therefore, if you're growing more yeast in a very high alcohol fermentation, you will get more of them. So they can be controlled, but they can't necessarily be eliminated. So esters and higher alcohols are fairly key components. Then you have aldehydes and ketones. The most famous aldehyde you'll have heard of is acetaldehyde, which is up here on the top right of the slide. And that's a byproduct of yeast fermentation. We'll talk a little bit more about it in a second. The only ketone you'll get in your fermentation is diacetyl, which you definitely will have heard of. Then there are numerous sulfur compounds produced by brewing yeast as they ferment. Now, they don't tend to be there in massive quantities, but they're extremely flavor active. So esters are there in quite large quantities, but are less flavor active. Sulfur compounds, smaller quantities, but very flavor active. And then finally, you'll have the phenols. So we already spoke a little bit about phenols, but that's the phenolic off flavor that you get from brewing yeast. Yeah. So this is what an ester looks like. It's just a type of molecular linkage between atoms in a molecule, but they're very flavor active, and there's lots of them. So if you got a light lager, the main flavor component you're getting from that light lager is probably esters. People always tell you that they don't taste of anything. That's not true. They do taste of things. It's just a bit more subtle than getting hit in the head with a load of hops that you might get in an IPA or something like that. So they do have a flavor to them, and it's usually quite ester based. So light lager tends to be quite low in bitterness, very low in hop flavor, but the main characteristic is going to be slightly malty with some esters. And if you drink a light lager, and I would recommend that you do so at some stage, just to kind of have a go at experiencing the subtleties of these, you'll probably get flavors like this. So ethyl acetate is the most common ester in beer, and that's because it's formed from acyl coenzyme A, which is a metabolite that exists within yeast, reacting with ethanol. Ethanol being the most common alcohol produced in a brewing fermentation. That's the reason why that's the most prevalent ester. And it's quite solventy, but not unpleasant. So sort of like slightly nail varnishing removery, but if you think that, but way less intense. So like not like you're sniffing it out of the bottle, but you're holding it over here. You're like, oh, okay, there's a kind of a nice, almost fruity characteristic to that. So if it's there in excess, it can be probably slightly unpleasant. But if it's there in smaller quantities, actually quite nice. So that's the most common. And then the next one, which it's probably one that you would have heard of most, is the banana ester that's from wheat beer specifically. So it's there in very high quantities in a wheat beer, but it's there in all beer. So it's present in all beer. And again, if you have a light lager, think probably have your eyes closed. Think bananas. Think pear drops. Think nail varnish remover, and that's the main flavor characteristic you get from it. Coors Light is the example I use. I don't know if you get that in Sweden. No, it's produced in Burton-on-Trent. It's probably I think probably one of the biggest volume lagers in the UK, but it's very estery. It's very bananas. It's very kind of pear drops. It's actually quite nice. And you see, compared with some compounds, they have quite a low flavor threshold. So I'll go back to ethyl acetate. This is at the bottom here, the flavor threshold, and that's the point at which 50% of people can actually pick out that flavor in a beer. So you can between 20 and 40 parts per million. It sounds quite low, but compared with some compounds, that's actually quite a high flavor threshold. So it has to be there in quite a high quantity before you pick it out, but it is because it's the most common ester. This one, isoamyl acetate, it's actually got a lower threshold. So actually you get probably less of it produced, but because it's got a higher threshold, you can taste it more easily. So when you do taster training, this is one that I usually, I like to use because it's very obvious. You pick it out straight away, and it's a good example of estery flavors that you would get in beer. Now, there are loads of esters. Those aren't the only two, and I'm not entirely sure if I have any more slides on them. No, I don't. But these are acetate esters. You also get ethyl esters, which will be formed from the reaction of this ethanol and then other coenzyme A molecules. So actually, in this case, it's bound to an acyl group, but you get fatty acid esters as well. So ethyl fatty acid esters. The most common is probably ethyl hexanoate, which has kind of an aniseedy, pineapple-y, red apple type flavor to it, which is quite nice. We have a product called Nova Lager, which I'll go into later on, but it produces that ester, but only really above 16 degrees. And then it's quite obvious is that it's the most obvious of the three. You get it from that particular yeast. And it can be quite nice. Again, in smaller quantities, it's nice. You don't want too much, but a little bit is absolutely fine. So there's lots of things that you can do to control ester formation. I said you can't eliminate it entirely, but you can play around with the work that you're using, the fermentation parameters that you're using, the strains that you're using to control ester production. So we already spoke about high gravity. So high gravity, high ethanol fermentations, there's always going to be more. So there's a little plus sign there, which just signifies that it produces slightly more esters. One of the things, and people aren't necessarily entirely sure why this is the case, if you actually use some additional glucose in your work, you can actually increase ester production. And it's not clear quite why that's the case, but it does. So you can add 2% glucose or something to the kettle if you were trying to get that up. If you put pressure on your fermentation, you actually reduce the esters being produced. Now that's a consequence of the fact that a lot of these reactions that take place within the cell are decarboxylation reactions. And if you put pressure on a fermentation vessel, you get a higher level of dissolved CO2, and therefore that reaction doesn't take place at the same rate, so you can get less in that way. And that's been used a lot by homebrewers in recent years, where they don't have very good temperature control. One of the things that they can do to influence ester production is actually pressurize the tank, because that's easier to do than control the temperature of the tank. High temperatures just favor yeast growth, and these are all things that come from yeast growth. So if you've got more yeast growth, you get more ester production. So that's the reason that high temperatures can do it. And then wort aeration can actually reduce it. So if you put a bit more oxygen in the wort, you can actually reduce your ester production. And then the yeast strain. So we saw with things like English ale strains, it can vary quite widely within the strain. Wheat beers produce a lot just genetically. So choosing the right strain will help as well. So if you're looking for something really clean, get a very clean fermenting ale strain, maybe like the Nottingham or something like that. If you're looking for something more estery, try a more traditional British ale strain and you'll get more esters from it. Yeah. Aldehydes, two most common, they're quite flavor active. Acetaldehyde, which I think I have a slide on, we do. But also oxidative flavors. So if anybody's ever had like papery cardboard from their beers, that's trans 2-nonenal, which is also a type of aldehyde. So you've got these flavors coming from aldehydes. Acetaldehyde shouldn't really ever have a problem with, but it does happen. So there's lots of reasons that it could happen, but one of those would be using healthy yeast. So this is a flavor that is metabolized by yeast into ethanol. So if it's there in excess and the yeast is healthy, it will actually reduce it down. If the yeast is not healthy, it's not very vital, it's not very viable, you might find that it doesn't. And at the end of the fermentation, that flavor will stick around. And it's one of the reasons that beer prior to maturation used to be called green beer, is because it had that apple green flavor to it. The other flavor it's quite often related to is emulsion paint. So if you ever do like painting, that's got like sort of weird acetaldehyde flavor to it. The other thing that it can come from is aerobic bacteria. So if you're making kegs, cans, bottles, you're less likely to have contamination with aerobes or bacteria that like to grow in a oxygen-rich environment. If you're making cask beer, it's going to have oxygen in it from the moment that barrel is tapped and it's poured in the pub. So if you're getting that green apples flavor from cask beer, let's say you're in London and you're in a slightly dodgy pub and they've left that beer on too long, that's what you'll get first. You'll get that green apple flavor. And then if I go back to this slide up here, you'll see this pathway on the right here. You have ethanol, acetaldehyde, and then acetic acid. And that's what those bacteria are doing. They're oxidizing ethanol. So ethanol's in the product as alcohol. They're oxidizing it to acetaldehyde. So the first sign of that beer going off is it starts to taste like acetaldehyde, starts to taste like green paint and apples. And then the second sign is it tastes like vinegar mixed with green paint and apples. It's not very pleasant. But it's a very obvious sign that cask beer's been on too long. You know your cask beer's been on too long if it starts to taste like that. And you don't often find it, but I do remember going into London during the COVID pandemic when they weren't selling enough and having one and being like, wow, that's awful, and taking it back to the bar and he said, yeah, it is bad, isn't it? So he knew. But it's a very, it's very specific to kind of cask beer production, that sort of acetaldehyde flavor. So things that you can do, ensure you've got good yeast viability and vitality. So yeast nutrient, make sure you're using good yeast. Make sure you use the right pitch rate, so you don't have enough yeast there. You might find that it doesn't reduce it down in fermentation as well. Appropriate vessel residency times, just leaving it long enough. So don't rush it through. Make sure that flavor is gone before you go to the next stage. Fermentation temperature won't make too much of a difference, but if you are going very hot, you might find it starts to build up a bit more before it reduces down. And then sanitation and cleaning. So particularly in the case of cask beer, that would be sanitation and cleaning in the in the pub cellar. So that's probably where that contaminant's going to arise, is actually in the air in the pub cellar. Not necessarily from the brewery, although that's possible. But the brewery environment tends to be more anaerobic, whereas the pub cellar definitely more aerobic. And I don't know if you've ever seen traditional cask production, but they tap the barrel, so they've got the tap in the base, and then the top they just drive in a wooden peg, so a porous wooden peg, and anything's getting through that. So some more modern pubs will actually put a peg with CO2 supply into it, which is cheating slightly, but does work and maintains your beer for much longer. So the ketone we're going to talk about is actually a diketone. So it's got two ketone groups, circled in green, is diacetyl. Now, all yeast strains produce diacetyl. It's very flavor active. So you can see the thresholds in parts per billion rather than parts per million. So if there was a billion people in the room, you'd be able to see 10 to 40 of them, which is, pretty sensitive as far as your nose goes. And some people like this flavor. If it's present in a very high level, I find it unpleasant, but I do actually quite like it in low levels. But it's kind of a toffee, popcorn, butter sort of flavor. And it's produced while the yeast is trying to make certain amino acids. So the yeast is trying to make an amino acid called valine, and it makes this chemical called alpha-acetolactate, which is then produced in excess and expelled from the cell. And then that chemical actually breaks down into acetyl within the wort. So it's produced within the wort. And then if you've got healthy yeast, they'll actually reduce it back down again. So they'll reduce it first to a chemical called acetone, and then to butanediol, and that chemical is flavorless. So if you have healthy yeast and you have enough of it, you'll actually produce the diacetyl during fermentation. There will always be a peak, and then it will come back down again after fermentation to a point where you can't taste it anymore. So you'll go below flavor threshold, and then you'll pass your diacetyl test, and you can move to the next stage. So one thing, if you were doing lagers, you tend to go with a diacetyl rest. You would raise the temperature towards the end of fermentation, and that would help the yeast to reduce down the diacetyl faster. In the case of an ale, because you're producing at a warmer temperature anyway, sometimes your diacetyl will be in spec at the end of fermentation, though not always. You can also cheat. So there's an enzyme that you can use, which is called ALDC. And it just catalyzes the reaction from alpha-acetolactate straight to 2,3-butanediol. So you miss out the diacetyl production step. You don't create it at all. Some people think that you can use this enzyme to remedy this situation. So if you've got diacetyl, you can throw it in and then you'll reduce your diacetyl, but it doesn't work like that because it catalyzes the reaction of the alpha-acetolactate to the butanediol. So if the diacetyl has already been produced, it's too late. You can't use an enzyme to reduce it. So if you're going to do it, you have to do it at the start of fermentation. That enzyme is also quite pH dependent, and your pH falls during fermentation. So actually the pH at the end of fermentation is not optimal for that enzyme to work, whereas it is at the start of fermentation. It can be a useful tool if you're trying to make lager quickly, but just make sure you add it at the start of fermentation. And that's pretty much what I just said in that slide. So we'll move on to the next one. So monitoring fermentation, we don't have to go into a lot of depth in this, but it's quite often different depending where you are in the world. So in the UK, we tend to use the specific gravity. Most of Europe and the rest of the world might use the Plato scale, but there's also the Balling scale, cider and fruit wine, they might use bricks. If you're a fridge engineer and you're testing the density of your glycol in your cooler, you'd probably use a different scale. You might use Baum. So these are all empirical measurements of density. So essentially they're all the same. It's just a different scale. And you could use a hydrometer with different graduations for each different type of scale. Or you could use something a bit more fancy like that DMA 35 from Anton Parr, and it would do the density for you by vibrating it. So it's, there's lots of ways to measure it. But the reason you measure it is because you're looking for your stages of fermentation. So at the start of the fermentation, the yeast can be quite slow, call it lag phase. And at that point, the yeast isn't actually taking in much in the way of sugar from its surrounding environment. It will be using intracellular sugar, and it's starting to create enzymes that it needs to break down the sugars in the fermentation. After that happens, the first thing you'll notice is a pH drop. So when you start fermentation, the first thing that you notice tends to be a pH drop rather than a gravity drop. So let's say you brew on one day and that brew is in the fermenter by, let's say, 4 p.m. Let's say you come to that tank the next day and you test the gravity. The gravity might have dropped by 1 or 2 points, maybe, depending on the yeast strain you're using, how much you've pitched, but that pH will have dropped quite a lot more. So the first sign of problems in fermentation would tend to be the pH. So if the pH hasn't dropped, you know there's a problem. So I would always look at pH first. It's very important to measure pH. And pH meters are a real pain in the ass. The finicky things, they tend to go out of calibration very easily. So I would like to calibrate your pH meter every morning. And then you know that at least it's accurate, because they do wave around quite a lot if you don't look after them. And breweries aren't ideal places for them. They tend to break quite quickly. I used to have one that was made of titanium for wastewater. It was really good. So yeah, if you find one of those, fantastic. So look at your pH, but then also look at your fermentation as well. After your lag phase, you'll get log phase, and that's when the yeast is growing, taking lots of sugar, fermenting very efficiently. And then towards the end, it will start to level off. And generally, I was speaking about these glycogen reserves. At the end of fermentation, the yeast is starting to build up those glycogen reserves because it's entering a state of starvation. And then it will flocculate, drop to the bottom of the tank, and it will have those intracellular carbohydrate reserves to sustain itself while there's no food in the local environment. At that point, the temperature will tend to drop as well, because during fermentation, it's an exothermic reaction. You'll get heat produced towards the end of fermentation, and that starts to die off, and the temperature will tend to drop as well. So all those things are what you need to look at. And I actually am a big fan of pH. I think pH is probably one of the most important things to look at. NPH should be quite low. So it should be in the region of 3.9 to 4.3. If your pH is 4.6, 4.7, 4.8, as it can be in like hazy IPAs, I would like to adjust it. It's not ideal to have it that high. And the reason is it makes it more susceptible to contamination. So bacteria don't like to grow at low pHs. Below about 4.6, they start to get a bit grumpy. I mean, some can, but not all will. But yeah, pH low. I think in recent years, there's been probably a tendency to have the pH slightly too high. So I would keep it nice and low. We can talk about fermentability. So this is attenuation, something that's widely misunderstood within brewing. So people always want us to attribute a level of attenuation to a yeast. And in practice, the level of attenuation in a particular beer or wort is much more related to the wort than it is to the yeast. So most brewing yeasts will ferment maltose, maltotriose, and glucose, as we've seen. And in that case, they're probably going to ferment anywhere between 75 to 80% attenuation, but it will very much depend on the wort. Something that doesn't ferment maltotriose, yes, it will ferment less, so it will attenuate less, maybe 65 to 70, but it will always be a broad figure, because the level of attenuation is much more dependent on mashing, on grist, than it is on yeast strain. So I could give you a broad guideline, but I'm not going to say it does 83, because it doesn't mean anything. It doesn't do 83, it does whatever it does in that particular wort. But every brand that you have, if you're making that brand over and over again, should have a consistent level of attenuation. So it should always be the same. So if it's not the same, something's gone wrong. And where can you look at to understand why that was the case? So I always ask first, was it me? Because I've been a brewer and I know that I'm not perfect. So was it me? What went wrong? Did somebody forget to put on the oxygen bottle and therefore the yeast didn't have any oxygen? Did somebody forget to pitch the yeast? Did somebody not pitch enough yeast? Was the mash temperature wrong? There's so many different things that can affect it. Then you could say, was it the maltster? Because as brewers, we love to blame maltsters for things. It might be their fault. It may not be their fault. And then finally, was it the yeast supply? Because it could be as well. There could be a situation where your yeast is not as viable as it should be. And therefore, it would probably tend to stall high. And then that could cause a problem as well. So there's lots and lots of different things that can affect attenuation, but it should be the same across your brands. If it's not the same, if your same brand is not the same every time, something has gone wrong somewhere in the line, and that's what you should be looking at. So that's one of the key things in your beer specification. If you're looking at that brand, pH should always be the same, attenuation should always be the same, the alcohol content should always be the same. And if you've got those three right, you do quite well. So that's one thing I would look at. There's lots of things that can affect it beyond that. So wort composition, we kind of already went into that a bit with mashing, but for instance, right now in my fridge I have a bottle of beer from a brewery in London that I collected yesterday, which I'm sending off for analysis, and it has stopped fermenting at 1020, and they don't know why, but they produced a light lager, so they used 25% corn, which is a raw material that doesn't have a lot of nitrogen in it, so maybe it doesn't have enough nitrogen. Yeast require nitrogen to ferment. Corn also doesn't have any enzymes in it. So when they mashed it, was the enzyme level sufficient across the entire grist in order for it to ferment properly? So what I've taken the sample for is I'm going to send it off for fermentable sugar analysis, because I don't know whether the problem is that there's not enough nitrogen or whether the problem is that there isn't any sugar to ferment and it's just stopped because there's nothing left. I suspect it might be both. But in the case of a nitrogen deficiency, you can probably fix it because you can add yeast nutrient. So you can add yeast nutrient fermentation. If that's causing the fermentation to stall, it might pick up again. In the case of fermentable sugars, you could add an enzyme, but it may or may not help depending on what the problem is. So for me, it's valuable to know what the issue was. So I'm going to send it away and just find out because I want to know. I'm curious. But it's just an example of what happens. And if you're doing something a bit out there, you really need to consider all the impacts that it could have. Other things that can affect it, so temperature. Fermentation obviously goes faster at higher temperatures. So you might have heard of these Kvike strains that ferment at silly hot temperatures. They rip through fermentation because it's so hot. Chemical reactions take place faster at higher temperatures in general. So that's the reason for that. Pitch rate can improve your fermentation performance. So if you pitch more yeast, you can find it ferments faster. If you pitch too little, you might find it stalls. We've already spoken a little bit about the downsides of over-pitching over and over again. But I'd rather go on the high side than the low side, because if you go on the low side, you can actually end up with a problem where you have to ditch the beer. If you go on the high side, yeah, you might find that you have to replace your yeast culture more rapidly. Wort aeration. So again, that's one. If you don't aerate enough, you might find that it stalls. And I have experienced this where somebody forgot to put the oxygen bottle on, and it's very obvious. You know, it ferments, and where that beer would normally ferment and stop at say 10, it would stop at 20 or 18, and nothing would get it going again. And the reason is it just didn't have enough oxygen. It didn't produce enough sterols. The cell population isn't healthy. If you pair that with a low pitch rate, perfect storm, and it's just not going to work very well at all. Yeast strain. So I think I mentioned earlier that some strains don't ferment maltodextrin as efficiently as others. So they will probably still ferment it, but they won't ferment it as efficiently. Some are just faster than others. So some might have a slower finish to the fermentation. Maltodextrin transport is also pH dependent. So if the pH is wrong, they'll find it harder to ferment that particular sugar. And then yeast health as well. So if you've got yeast that's not very happy, yeast that's got poor vitality, poor viability. Viability is easier to measure than vitality, by the way. Viability is, live cells. Vitality, they might be alive, but they might not be fermenting very well. So you can think of it as a young vital cell would be like a teenager. A not very vital cell might be like an older person. So they might not be as efficient at moving around and metabolizing. So both of those things can affect fermentation performance. Flocculation is an interesting one. So flocculation is the ability of yeast to stick together at the end of fermentation and drop to the bottom of the tank, which is quite an important characteristic if you're repitching, particularly, or if you're trying to produce beers that are bright. I appreciate that's less of the fashion these days, but it can cause you no end of problems if you are trying to create bright beer and it isn't fermenting properly. One of the things that can inhibit flocculation is actually the presence of sugars. So if you have a fermentation that's stalled slightly high and there's a residual sugar left over, it can actually inhibit flocculation. So you can then be in a problem where it's also really hard to filter, or it's really hard to centrifuge. And these genes are also quite prone to mutating. So if you are repitching over many, many generations, you might find that your flocculation ability changes, and that might be the point where you need to refresh the strain because it just stops fermenting, so it stops flocculating efficiently. But it is quite a complex mechanism, so there's quite a lot involved. So it does depend on calcium ions, it depends on pH. So it's quite a complex mechanism. But it can be a sign that something's gone wrong if it's not flocculating correctly. Calcium is probably the easiest thing to get right, because if you have enough calcium in your water and it gets carried through from your mashing process, that's never going to be deficient. The only place where I think that could be very deficient would be very, very soft water. But it'd have to be really soft. I think you're unlikely to have too much of a problem with it. What's the water like in Stockholm? Is it hard, soft?
No, it's quite low in both calcium and magnesium. So
okay, so you may need to increase it.
yeah, we always do. Do you have a account that you recommend? How much calcium do you need in ppms?
Are we testing now? I think it's probably about 100.
Yeah.
Thereabouts. Yeah. I only ever brewed in very hard water areas because the UK tends to be quite hard until you get up north, and we always had the opposite problem, just getting it out. Well, it was so hard. We were on river water as well, so it used to change all the time because it'd spike every time you had rainfall. So yeah, I'm really lucky really. Soft water is great.
We take surface water from the big lakes, and we usually do that in the most of Sweden. So we always have quite low iron count in our water. So it's very good for brewing because
Yeah, great. No, yeah, I always got struck down with very, very hard water. So in and around London, it's why London was famous for stout production, because it's acidic and it drops the pH. Right. I don't know if you want to have another break or not, or I can go into this, because this is more on the kind of how we produce it, how we innovate, that sort of thing. So it's sort of almost like a different section.
yeah, we can take maybe just 5 minutes now. Then
Yeah, 5 minutes works. Yeah.
10:40 or 9:40, depending where you are. So
Yeah.
again, so
Okay. Go again. So talk a bit about propagation and management of yeast, but then we'll look as well about how we do it in plants, and something on kind of how we innovate on strain as well. So I might do the first bit reasonably quickly. So you've got options really. So this is probably a comparison of dry versus wet, and some of the differences between them. So I sell dry yeast, but I'm also very aware that people use wet yeast as well. So I don't really have a opinion on what's better or worse. I think they can both be used in the same way in different circumstances. And the dry yeast that we have is so repitchable that after generation 1, it essentially becomes wet yeast. So after that point, it's just a yeast cell. Differences at the start, you tend to have a high viable per gram of dry yeast. So because you've taken all of the water out, there's actually more cells within that gram than there would be of a wet yeast. The difference being that some of those dry cells will not be alive because they've been killed during the drying process. That isn't necessarily a problem because they can provide nutrient for the cells that are alive. So if you think about a human, everything that makes up a human is what a human requires to survive. So we contain all the essential elements that one would need to create a new human. Much the same with yeast. So some of those dead cells will provide nutrient for the live ones. Dry has a long shelf life, so you can keep it on the shelf for a long time. Even if you're a wet yeast user, it's probably something I would recommend to have a few pags lying around, because then if you do have a problem with your culture, you've got a backup, and you can keep it on the shelf, and it's not going to be a problem. You do need to keep it in the fridge, I'd say, not just on a shelf, in a fridge shelf ideally. Some of the wine strains are actually very stable in temperature, so you could keep them on the shelf, but brewing strains are a bit trickier to work with. As we spoke about before, there's less variety in the dry, and that's because they're not. You need to find something that's very good at surviving the drying process. With wet strains, that's not a problem. So it's never a problem. There's a price disparity between the dry and the wet. So some of the dry can be cheaper, some of the wet can be more expensive. But actually, you'll find that between the suppliers, that maybe isn't as true as it used to be. Both can be repitched, but both require you to have good quality control within the brewery environment. If you don't, neither are going to repitch particularly well. Dry, you don't really need to aerate it at the start of fermentation. Wet, you definitely do. If you don't, you'll have a problem. And with that in mind, the wet, you probably need to prop it up sometimes as well. So there's a limit to how much wet yeast your brewery can send, your yeast plant can send you, right? So if you've got an 80 hectoliter tank, that's probably right on the limit of where a wet yeast lab can send you enough volume for a fermentation. So you might need to do a step at the start to get that up to the right level of cells for a fermentation. In a case of dry yeast, it's actually quite easy to obtain enough for that fermentation. So where I do business with very large breweries, it would be if they're trying to do, like they're trying to do a new and interesting beer using a new yeast, but their yeast propagation plant is dedicated to their house strains or doesn't have the capacity, and therefore they'll be able to buy enough dried yeast to do that larger fermentation. The case of wet, if you're going to repitch it or you're going to propagate it, really better to have a lab. With a dry, I don't think you need to, but I would still recommend that you have something. I would always recommend that every brewery has at least a microscope, and ideally something more than that as well. So not essential, but I'd prefer to have something.
And how do you say about? I think if you have a brewing system that produce, let's say, 10 hectoliters, but you have fermenters that can take 20 hectoliters, is it good to brew one day and pitch for 10 hectoliters and brew next day and more 10 hectoliters again and pitch it, or is that a good
We would
way?
always recommend that you pitch the entire amount into the first turn, because at that point the level of nutrient and oxygen in the wort is higher, whereas the second turn it starts to be consumed. So I would probably do all the yeast at the start, and then when the new wort comes in, it's going to be actively fermenting, so it should be fine.
Oh yes, so you count for 20 hectoliters, but you pitch that in the 10 hectoliters. Okay.
Exactly. Yes, exactly.
Because I thought you can actually count for 10 hectoliters and it starts to grow, and then you can pitch.
I mean, you could probably do that. So if you pitched a small amount and then added loads and loads of oxygen into that first bit of fermentation, you might find that you get enough yeast cell growth in order to do the whole fermentation, but it probably wouldn't be best practice. I'd probably recommend that you do enough of the whole fermentation into the first turn. Yeah, that would be what I would recommend. So pitching dry, you could rehydrate it, but we've changed our recommendations on rehydration over the years because it isn't always necessary. Originally, when we came up with the rehydration method, it was when Lallemand hadn't really produced brewing yeast before. So that was something we did for our wine strains. And over the years, we've come to realize that the differences between a wine must and a brewer's wort are so great that you don't necessarily need to rehydrate when you're using a brewer's wort, because it's a much nicer environment for the yeast than a wine must is. So wine must is very low nutrient, very high hydrostatic pressure, whereas a brewing yeast, a brewer's wort has loads of nitrogen in it. It's quite low sugar, relatively speaking. It's not a harsh place for the yeast to be. pH is quite high. So we've done a lot of experiments doing side by side, dry versus rehydrated, and you'll find that there's statistically no difference between the fermentations. So unless you're doing something in really harsh conditions, say like a very sour beer or a very high gravity beer, there's not really any need to rehydrate. So we wouldn't necessarily say don't do it, but for many brewers, it can be a logistical problem. You know, you need quite large buckets and you need to stir them, and it can be a bit of a microbiological hazard. So I'd prefer to just dry pitch a bit with me.
Oh, that was interesting, because I brew a lot of sour beer. So I should actually rehydrate it when I do a sour beer, maybe.
Yeah, low gravity sour beer is like a slightly more hostile environment. I'd say, you know, if you're not having problems, then I don't think you need to fix them. So
Oh no.
if it works fine, it works fine. But it's one of those environments where the cell might not like it quite as much.
Yeah, but it's good to have in mind if you're starting to have a problem. It's because if you do really sour beers, if you maybe go under 3 or something, it can be.
One of the biggest problems we see with sour beers is terminal acid shock. So if you do like a spontaneous fermentation, and then you want to do a secondary fermentation in the bottle to create a fizzy product, sometimes the yeast just say no. And the reason is acetic acid. So it's quite toxic to yeast. So you might find a failure to actually carbonate because of that. So that's something to bear in mind. And the only thing you can really do about that is sort of slowly condition the yeast to very low pH and then dose it in.
Oh, great.
So usually pitch by weight with dry yeast rather than cells, and it'll give you a rough kind of level to pitch at. Although you can calibrate that if you want. So you could take your dried yeast and say, look, I got from this dried yeast this level of cells in the fermentation, and therefore I'm always going to pitch at this weight. But we tend to recommend fairly broad guidelines between 50 and 100 grams per hectoliter. So 50 would be if you're doing a standard 4 or 5% beer. 100, you might get up to like 7-8%. Beyond that, if you're going really high gravity, it's actually better to go beyond 100. So if you're going up to like, I don't know, like a silly 14% stout or something, then I'd actually recommend you go even higher. So yeah, you would vary those pitch rates depending on the style of beer that you're doing. So yeast slurry, you probably pitch again by weight. I would pitch by weight if I could. That would be the best way to do it. Because if you're trying to measure the number of cells in a liquid slurries contain quite a lot of CO2, and it's very hard to get an accurate volume of that particular yeast. So if you pitch by weight, you know at least you've got a gram's worth of yeast in that tube that you've taken. And then you can define your pitching rate. There's the old German rule of thumb, which is 1 to 1.5 million cells per hectoliter per degree Plato. But it's a broad guideline. You know, I wouldn't necessarily stick to it. You might find that you need more. You might find that you need less. So you'd work out the number of cells that you need for the total fermentation, divide by the cell density of the slurry that you've got to work out the weight of yeast that you require. And then you would aerate it as well. So when you pitch it, you have to aerate it because otherwise you get your stunted fermentations. So to work out your live cells in your slurry, you would use something like this. So this is a counting chamber, and you would dilute your yeast first, because if you try and put yeast slurry in that, it's going to look like a mess. So you need to serial dilute it and stain it with a chemical called methylene blue. And that's a chemical that will be ejected from live cells, so they'll kick it out of the cell. But in dead cells, that won't happen. So you can work out a percentage viability of the culture, and you can use that in conjunction with the total number of cells in the culture to work out how much yeast that you need to pitch into the fermentation. Caveat is that this doesn't work for dried yeast because the cell structure after the drying process is slightly leaky, so they'll turn blue even when they're live. So it doesn't really work very well. You can use it, if you're mid-fermentation and you've used dried yeast, fine, no problem at all. You can use methylene blue, but at the start of fermentation, it doesn't really work. So when we do our viability counts for dried yeast, we'll actually do plate counts. So we'll serially dilute the dry yeast and put it on plates, and then we'll use a plate count method to work out the viable cells that were in that amount of yeast that we plated onto the plate. So most modern yeasts are used with the cylindrical tanks. More traditional ale strains, you might be able to use top croppers. We talked about suction lances and top cropping in UK breweries. Not many of those strains can be purchased dry. You can get them wet, but most modern strains are basically designed for bottom cropping from cylindrical tanks. You can store that yeast in the cone of the tank. Particularly in smaller breweries, that's not too much of an issue. Or you might have specific vessels for it, or even buckets. So when you do store it, these are kind of things that you should consider. So the temperature should be quite cold. If it's very high alcohol content, you might want to reduce that down a bit, because the yeast likes to sit around in 10% alcohol. It doesn't live very long. Ideally, you don't want to keep it very long. So one of the things with repitching yeast is you kind of need to hit a critical mass of production where you're actually repitching that strain continuously, because you don't want it to leave it to sit too long, because it will be deteriorating the whole time it is. So 2 days to a week is fine. I say that when I was at university, we used to take yeast from the local Bass brewery and we used to use it after like 3 weeks. And the viability would be about 50, and you just add double the amount. It's not great practice, but it can work. If you needed to, it could work. So this is more of a storage consideration with large breweries. So you think about the kind of the surface area to volume ratio of a cone. In a small brewery, those cones are quite small, and therefore the chilling on the cone is cold enough that it can reach the center of that cone. But yeast is actually quite insulating. So if your cone is, let's say, as big as my living room, you might find that the yeast within the center of that cone starts to heat up and starts to produce its own heat, and therefore the cells don't have any food and they start to break down and degrade and solidify. So cones can be stored, yeast can be stored in cones for smaller breweries, but for larger breweries it really isn't viable. You would need to store it in a yeast storage tank and keep it agitated. You also get this layering effect in cones, so you get the old and dead cells falling to the bottom of the tank first, and they should always be purged before you take that cone. And then at the top, you might have very young mutated cells, so they've lost their ability to flocculate as well. So they'll be kind of the top layer. So ideally, you don't want to take the whole cone. You want to take the middle healthy bit. But it is quite hard in practice to do that. You don't have glass cones. You can't be testing every fraction that comes out of it. So that's the theory. But in practice, just get rid of the first bit and don't take too much. I think it's probably the best way to go.
I can understand muted cells, but what is bad with very young cells?
They're smaller.
Oh, they're smaller.
Yeah, they're smaller, so they haven't grown as much, so they can be near the top. But young's not necessarily a problem, but young mixed with mutated is more of a problem. Yeah. So there's some ideal storage solutions. So when I was brewing, we had a tank on load cells. So calibrated tank on load cells with a what kind of pump was it? It was one of the air pumps, diaphragm pump. and you could pitch on that basis, and the pumps can handle slurries, right? So they can pump yeast no problem. So you'd use that or a peristaltic pump, which kind of squishes a hose as it goes around and can pump solids as well. And we had an agitator in the tank, so you could pitch on that basis, which is good. Still quite a lot of user error involved in that. You can get a thing called an Aber monitor, which actually pitches based on capacitance. So the technology basically is that you can charge the cell membrane of a live cell, but you can't charge the cell membrane of a dead cell, and therefore that sensor can work out the amount of live cells it's pitched in line. They're very clever. They do a craft one now, but I think it's a craft size, but maybe not craft price. So they're quite expensive, but very innovative technology. And actually, a lot of the large breweries do use these now for consistent pitching. On a budget, and even not so much on a budget, a bucket in the fridge works perfectly well. There's a large brewery in the UK called Y Valley, big cask brewery, and they have a walk-in shipping container filled with dustbins, and they suction lance into the dustbins and put the dustbins in the fridge, and it works fine. So you can go really techy, or you can go low tech and clean. Just make sure you clean those buckets really well. Acid washing, I'm not sure if that's a UK thing. It probably is a bit of a UK thing. So acid washing is when you're maintaining yeast cultures for a very long time, let's say 30 years or something, and you're top cropping, or bottom cropping, works for both. But you might get problems with trace bacterial contaminants over time. And one thing that you can do about that is take your yeast slurry and drop the pH using food grade acid to like 2, pH 2. And for a short period, the yeast can withstand that, but the bacteria can't. So the bacteria die. And therefore, you've kind of cleansed your yeast culture, and you can continue repitching that yeast for future generations. Important to note, it doesn't work for wild yeast. So the wild yeast will be like quite happy at the pH 2. As will the brewing yeast to a degree, but the bacteria will die off. So it can be quite a useful technique. And let's say you're a big cask brewer, and one of your kind of unique selling points is that you'll have a yeast culture that's been going for 100 years. You don't want to throw it away and refresh it. It's probably a mixed culture. It's probably quite interesting, but you also don't want there to be a large level of bacterial contaminants. This is the solution. This is what they would do, and do still. The large breweries tend to not do it. It's kind of like a large regional technique, I would say. If you're like Coors or something, you won't be doing this sort of technique.
What kind of acid do you use to go down to?
I think it's higher food grade hydrochloric. These, I think, food grade hydrochloric. But you could, in theory, use anything. Yeah, I think probably a strong acid you have to use less of it. So we've got a question like lactical phosphoric. Yeah, you could use those as a weaker acid, so you probably have to use slightly more. But hydrochloric would be the one that is used for it. Food grade hydrochloric acid. Murphy and Son sell it. So bit on yeast propagation supplies. This goes into how we make yeast, and then also a bit on innovation. So this is our yeast factory in Vienna. It's a lovely building, and it also happens to be on the same site as the Ottokrona Lager Brewery. So behind it, you've got this big lager factory, which is those large tanks that you can see. In the foreground in red is the yeast factory. And kind of right-hand side, you've got these molasses tanks. So dried yeast is grown on molasses, so that's the sugar source for growing the yeast. But the far right is a former molasses tank that they've converted into a brewery. So they've cut the steel out and they've made windows, and the top floor is a brewery. Middle floor is fermentation, and the bottom floor is a little kind of tap room and bar. So it's quite cool. Yeah, it's a cool place to go. The brewery's much more exciting than the yeast factory, if you ask me. But yeah, that's where we make all of our brewing yeast, bar a few strains. The Belle Saison, which is made in Paso in Germany, and then the GMO strain, which is made in Brazil. So most of our brewing yeast comes from there. So when we start, our propagation process is very much, in the first instance, like a large brewery would do it. So somewhere like Carlsberg would probably have a central strain bank where they keep all of the strains in liquid nitrogen, and then they send them off around the world to the various breweries where they're propagated up and turned into large amounts of yeast. Same for us. So all of the brewing strains, the wine strains, the baking strains, everything, they're all held at the Lallemand Yeast Culture Collection, which is in Montreal. And there's over 200 different types of brewing yeast there, stored on slants. in liquid nitrogen, and then when they're sent out to the plants, they'll be stored in a standard freezer at -80. Interestingly, something that we've done recently in our research is that even at -196, which is where we keep them, the idea being that you get no mutation at that temperature, even at -196, you can get genetic mutation. It's just very, very slow. So there's no way to perfectly preserve a strain, albeit, there's going to be very little changes. There will still be some changes, which is fascinating if you ask me. Where do our yeast come from? We've got strain libraries like our own, so Lallemand Yeast Culture Collection. Then we might look at the Doman's Academy, so we've licensed some strains from there. We don't have anything from the NYCC, that's the National Yeast Culture Collection in the UK. And then there's Weihenstephan in Germany as well. So there's loads of those. Lots of different places around the world have strain banks, and you could take some of those and grow them up and market them as a new strain. The downside with them is they tend not to be very well characterized. So they'll probably just say brewing strain deposited in 1942, and you won't know anything more about it. So at that point, you have to take it into your lab, do all the work behind it, work out kind of flavors it produces, how it performs. And then you could maybe get to the point where you were to launch it. So if you were to do that and it was an unknown strain, you might be like 2-3 years before you could come out with a new one. We also work with research partners. So we have people that come to us and say, hey, we've got this really cool strain. We think it would work well in your collection. Do you want to launch it? There's a story of Philly Sour, which I'll talk about, but Philly Sour is one of those. Then we might work with partner breweries. So our Verdant strain was the yeast strain from Verdant Brewery in the southwest of the UK. We also produce some strains for like very large guys. So ABI, we do some proprietary yeast for them for certain brands that they aren't able to grow the yeast for. So we might take some of their strains. We wouldn't market them because they wouldn't let us, obviously, but we produce it for them and others around the world. And then we might work with commercial biotechnology companies as well. So we have a very close relationship with a company called Renaissance, who are experts in yeast breeding, and we work with them to produce new strains as well. So types of yeast discovery. Bioprospecting is quite a cool thing. So this is the story of Philly Sour. But Philly Sour is a strain that was isolated from the environment, which is cool because there's loads of different strains out there in the environment, but most of them aren't very useful for brewing. And it's very hard to find something useful. So, just going out there and in your garden, the likelihood of finding something that's good for a brewing environment is pretty low. But if you were to find something unique that's good for brewing, you wouldn't have to genetically modify it, which is a good thing too. So we have one strain that was produced using a bioprospecting technique, and that is our Philly Sour. So that was isolated first by the University of the Sciences in Philadelphia, and that was isolated in this sort of park graveyard type thing on the top left, from an oak tree, I think. So the original code name they had for it was GY7B, which was Graveyard 7B. And they just did it by sampling. So they were going out and swabbing, site sampling different trees and things, and they enriched it and they isolated a single strain from that. Following that, they did a bit of DNA identification. So you can isolate DNA that comes from the ribosome, which is very specific to yeasts. And then you can compare that with online databases to see what you found. And in this particular case, they found lots of things, which is quite interesting. But they also found in red human pathogens, which is one of the reasons why this technique might be a little bit tricky, because if you're going out into your garden and isolating a yeast, you don't really know what it is. And there is a small chance that it could be something that might not be very good for you. Yeasts don't tend to pathogenize people very much unless you're immunocompromised, but if you are immunocompromised, it could be a problem. So yeah, it's just worth bearing in mind. But one of the strains that they did isolate happened to be Philly Sour, which is a novel Lahansia species, and not a human pathogen, which is good. And what's interesting about the strain is it produces lactic acid as it ferments. So weirdly, for a strain isolated from the environment, it actually ferments maltose and maltotriose and glucose, which is unusual because that wouldn't be normal. There's no genetic pressure for it to do that. So the fact that it does that is quite interesting. But it also produces lactic acid from glucose at the start of fermentation. So during fermentation, you get this first kind of stage where you get lactic acid production, and then it kicks into alcoholic fermentation. You get alcoholic fermentation as well. I'm not sure my slides got jumbled up, but there might be something more on that. But if there isn't, I'll just describe it quickly now. But one of the one of the impacts of that is because you're using glucose and creating lactic acid, is you actually get slightly less in the way of alcohol production, because some of that sugar is going into lactic acid production rather than alcohol production. So it's not a lot, but it's a little bit. So it's a very good strain for sour beers. You don't have to do a kettle souring step as you would if you did a lactobacillus bacteria. And it's actually very safe. So it's quite a wimpy strain. It only produces lactic acid from glucose. So even if you had a contamination problem with it, as soon as the glucose was gone, there'd be no lactic acid produced. And it's so slow compared with the brewing yeast that it would never really get a chance to get going. It likes the warm. So it has to ferment between 25 and 27 degrees, whereas a brewing strain would be at 20 or lower if it was a lager yeast. So it's very safe, and doesn't really stick around either. It's quite easy to kill. Right. I hope there isn't another strain coming up and I'm out of order. No, I'm not. That's good. Okay. So this is our kind of more modern way of strain production. So whole organism selection is what we've called it. But there's two different ways in which you can do it. So there's adaptive laboratory evolution, which is where you force a strain into evolving in a certain way by putting it in nasty environments. So we have a strain produced at a lab for us, well, one of our labs in Tallinn in Estonia. And they've created some interesting wine strains for us that produce naturally less ethanol. And the reason for that is climate change means that there's too much sugar in grapes, which means you get too much alcohol in the wine that you eventually produce. So what this strain does is it produces glycerol as well as ethanol. So it will reduce the amount of alcohol you produce, not by a lot, but up to 2 or 3%. So at the end, the wine is at the correct alcohol content. And the way that they did that was they took a wine strain and they grew it for, I think, hundreds of generations in very high levels of salt. And then it starts to produce glycerol as a stress factor. And eventually that becomes ingrained and becomes a genetic change. And it's then able to do that and produce less alcohol content. So that's one of the things that you could use that particular technique for. The one that we've used more recently in brewing is selective breeding. So that's like the lager yeast when it was first made, when it first came to be. You actually force the strains to sporulate and then breed together to create new strains. So you can introduce characteristics into a strain that are beneficial, but it's not genetic modification. So it'd be like breeding dogs rather than inserting foreign DNA into that strain. So it's something that you can do and you can legally do in Europe, whereas GMO, you can do pretty much anything, or you can do a lot of things, but you can't do it in Europe. So there's not many places that you can use GMO yeast, at least for food production. You can use them for ethanol production, for distilling and, well, not distilling, sorry, for fuel ethanol all over the world, but for food you can't. So we work with a company called Renaissance, who are in Vancouver, and they are experts at breeding. We've done a few strains with them. So one of them is a Saison strain that cannot ferment dextrins. So it's got that Saison background, but we've bred out its ability to break down dextrins. But the most recent one that we've done is not this one, actually. We've done a low alcohol one recently, but the most exciting one from last year was this lager strain. So it's called Nova Lager. And it is a new type of lager. So at the moment, we've got type 1 and type 2. And the reason that we did it is because you've got a lot of microbreweries out there that aren't necessarily able to hit some of the conditions they might require to make really quality lager. So they might not be able to go to a low enough temperature. They might not have the time, the space for the long fermentations that you would need. They might not have the conditioning time to reduce the H2S production. And they might not have time to get the diacetyl down. So this is a strain that was designed to kind of hit those particular problems within smaller breweries. And some larger breweries have used it as well, actually, as well. So I've got a big group in Finland that use it for some stuff, which is exciting. But what we've done is really change the genetic background of the strain. So the top right here, you have the original wild ancestor of lager yeast, which is this Saccharomyces eubayanus strain. And then on the left-hand side of the strain, I've got the type 1 yeasts, and they are about 2/3 wild strain, so 2/3 eubayanus in their genetic material, and 1/3 ale yeast, which is the reason why they are not very good in brewery, and the reason why nobody uses type 1 yeast for anything. If you look at the type 2 yeast, so these are your classic lager yeast that everybody uses all over the world, you can see they're about 50-50 on the genetic background. So they've got 50% of their genetic material coming from the wild ancestor of lager yeast and 50% coming from ale, which is great. They make nice beers, but they're still quite slow. They're still like cold temperatures. They produce lots of H2S. So what we've done is actually breed a strain that is only 1/3 of the wild type ancestor, and so 1/3, 1/4, and then 3/4 Saccharomyces cerevisiae. And at the same time, we've adapted, inbuilt an ability not to produce H2S. So as the strain ferments, it can't produce H2S as well. So it hits all those points. You know, it ferments slightly warmer because it's got that greater amount of cerevisiae genome in there. It doesn't produce any H2S, which means it needs less in the way of lagering time. And it also has quite a low diacetyl peak. So I think I've got some slides on that. Well, this is fermentation versus a standard yeast. So yellow is the Nova lager, and orange is our type 2 standard lager yeast, and you can see it ferments much quicker. So it's getting down to the end of fermentation a lot faster. But it also has, this is the interesting slide. It has a much lower diacetyl peak. So the right-hand side of the slide, you have Diamond, which is our type 2 lager yeast, and you can see it produces an awful lot of diacetyl during fermentation, which is normal for a lager yeast, but it also means it takes much longer to bring it down. Whereas the Nova lager is much more efficient at bringing in valine from the environment, so it has a much lower diacetyl peak, and therefore it finishes a lot quicker, mops up that diacetyl a lot faster. It also, this is where we've made a silly wort. So we've made a wort that's got very, very low levels of nitrogen in an effort to try and force the strain to produce H2S, which is the rotten egg flavor that you get from lager yeast. And you can see with the Diamond strain, which is the classic lager yeast, it's producing loads of it throughout fermentation, which would be a problem because it would be there at the end of fermentation, and you have to wait for it to either flash off, or you need to force it with CO2, or treat it with copper, find some way of getting rid of it. With the Nova lager, it doesn't produce any at all. So you've never got any H2S to worry about. It's genetically incapable of producing it. Now for some people, that's a problem because some people want a little bit of sulfur in their lagers. But for most, it'll actually taste really clean. So, you know, if you wanted a bit of sulfur, just use the standard lager yeast. If you're not wanting sulfur, I'd probably try the Nova and see what it does. But I wouldn't go above 16 because it produces the ethyl hexanoate. So you get kind of pineapple-y kind of flavor above 16, 17. It starts to produce a bit more ester. So go on the cooler side. But it's a really cool, innovative strain. Then we might also look at genetic modification. So genetic modification is a really interesting way of creating new strains, and something that's really big in the United States right now. So most of the big yeast labs in the States have at least one GMO strain, and there is an entirely GMO yeast lab in the United States called Berkeley. One of my colleagues actually works there now, and you can introduce all sorts of interesting characteristics. So you might be able to say, you can introduce an SDA gene, so the strain would be able to ferment dextrins, and you could use it to produce light beer. So the first ever GMO brewing yeast was actually created in the UK and did that in the mid-90s. So you could do something like that. You could create a strain that produces loads and loads of thiols, and that's something that's been quite big recently because thiols are really big subjects, and the kind of flavors that come from hops, very, very flavor active. But brewing yeast as a whole, in general, are quite bad at producing them. Some wine strains produce them quite well, but they only produce them well in a state of nitrogen repression. So in a grape must, where there's very little nitrogen, they produce a lot of thiols. If you then use those wine strains in a brewing environment to try and have the same effect, it doesn't work. So the only way that people have found to do it so far is to genetically modify them, to introduce pathways that can produce loads and loads of these flavors. We have one strain, which is called Cerevisiae, and we have a business called Muscoma, which is in New Hampshire in the United States, and they very big kind of research facility for us in many ways, not just GMO, but they do some GMO yeasts, mostly for biofuels, but I think maybe in their lunch break they play around with brewing stuff. So we've got one brewing strain from them where they have introduced a lactate dehydrogenase gene into the genome of that strain, and it's able to produce lactic acid in very, very large quantities. So you get a beer that's very, very sour very efficiently. And there's some downsides to that. It produces so much lactic acid that it's not very balanced. It's maybe a bit too much lactic acid. It also produces quite a lot less ethanol because it's producing so much lactic acid. The other thing that's not very good about it is it doesn't repitch because it produces so much acid that it actually sort of kills itself in the process. So it's not a very healthy strain. You could try and repitch it, but not for very long, because it would not be very happy. So they are thinking about doing a Sangiovese 2 that produces less in the way of lactic acid. But one of the things we have done is we've done some blending experiments. This doesn't display very well because it's not white, so sorry about that. But if you then blend the strain with other brewing yeasts, you'll find that it produces less in the way of lactic acid. So if you put in just the lactic acid strain, it produces loads of it. But if you say put 50% of that, 50% of a different strain, then you can start to temper it down and make it a bit more reasonable. GMO isn't perfect though, and we have had a strain in development for a number of years, which is designed to make a ketone that has a berry flavor. And it's weird in that you found that 50% of the time it worked, and they didn't really know why. So they've done all these brewery trials. We've produced the yeast, done trials all over the world with a view to launching it, and you found that most of the time it worked, and then sometimes it didn't, and they're not quite sure why. So you have to go back to square one. And start the whole process again, add a new pathway. So there's quite a lot to it. It's quite involved, but certainly fun and interesting. And I think probably a few years off yet, but at some stage Europe will probably come around to the idea. Not really, I think brewing will be the priority, but food security globally will probably require us to need to genetically modify things in order to make them more efficient and feed the world population, which is still growing. So I think it will change. At some stage, it's just a question of when. So dry yeast production, we've got some steps. So you already saw a picture of the brewery. I think there wasn't any pictures of what it looked like yet. So this is how we do dry yeast. So the first steps are very similar to a brewery propagation. You've got 3-step lab culture. So you've got your slant, and you go into like a little 20 ml kind of conical flask, and then up to a bigger one. And then you'll go into a thing called a Carlsberg flask, which I've got a picture of, which is I think about 20 litres. Then you'll go into the first stage of fermentation, which will then go to a separator. So you'll create a cream yeast, which will get stored in a cream yeast tank. You'll then use that as a starting point for a next fermentation, which will be the big one. And you can use that seed yeast a couple of times. So you might use half of it for one and half of it for another, create 2 batches. At the end of that commercial fermentation, you induce a stress response. So this is when you're creating that dry halos, which allows the yeast to survive the subsequent drying process. So you're having to force that yeast to create dry halos. And then something that people don't necessarily realize is because we're growing on molasses is a natural byproduct. It varies quite a lot in its color. So when you look at your dried yeast, you'll find it varies in its color from very, very pale to slightly darker. What we found over the years is that brewers look at the darker yeast and say, hey, there's something wrong with that. And there may not be. It might just be the molasses, but we try and control the color by washing it. So you'll put it through the separator and then you wash it with water, clean water, to try and reduce some of the color. So we have a scale, I think, goes from 0 to 5. And normally it'll come in somewhere like 2 or 3, but on occasion it might come in a little bit higher. So it varies in its color. After that point, it goes to a thing called a rotary vacuum filter. And that's where you spray it on the outside of a drum to which a vacuum is applied, and it sucks the water out of it. And the yeast is cut from the drum using a knife, extruded through a plate, which makes something that looks a bit like a spaghetti. And then that is sent to what they call a fluidized bed dryer, which is essentially a big tank with a perforated bottom into which they blow hot air. And at the start, the temperature is actually very hot, hotter than you would expect. So it's like 80 degrees. And the reason is because there's so much liquid in it. That liquid evaporating takes the temperature away, takes the heat away. So as the drying process proceeds, you turn the temperature down and down and down until a point where the yeast can't be damaged by it. It might be damaged to a degree because it always will be, but it won't be to a point where you just burn the whole lot, because that's the risk. And we have, I think we tried to produce brewing yeast in one of our big plants in Montreal once, and the drying program wasn't adjusted, and it just burnt the entire batch. Went in the bin. So it's actually quite a key in the process is that drying. It's actually quite complicated. During the actual commercial fermentation, that's quite complicated as well, because you have to ensure that the yeast isn't going for the crab tree effect. So you have to ensure that it's not fermenting. When yeast ferments, it doesn't create much in the way of biomass. So it's creating ethanol, but it's not creating that many more cells. We're wanting the yeast to grow aerobically. So we need to ensure that the sugar concentration isn't too high, that the yeast starts to ferment. and the oxygen isn't too low. So it needs to have enough oxygen, it needs to have not too much sugar, and it's quite tightly controlled. So you measure the ethanol in the exhaust, and the pH and the level of sugar in it. So there's quite a lot to it. We'll then bulk package. So we take the dry and we put it into 20-gram bricks. And that might go out to some larger breweries. Some larger breweries take 20 gram, 20 kilo bricks of yeast. Most smaller breweries will take 500-gram packs, so they'll be repackaged into 500-gram. This is a bit what it looks like. So top left is a slant from the Seaboard Culture Collection. Then you have the stages in fermentation culture. So I've got the proper volumes here. So 1 litre, so you go slant into 1 litre, slant into 125 mils, then into 1 litre, then into a 9-litre Carlsberg flask. Then you do a pre-culture, so you do a 100-litre step, then into a 10,000-litre step, and then the big ones are 1,000 to 1,500 hectolitre. And that's a small yeast plant. So you can see when I was speaking before about contamination, if you think of the number of replications you have there, you start with, let's say, 1 cell or very close to 1 cell, and you scale it up and up and up and up, and you've got billions and billions and billions of cells. If you have 1 contaminant cell at the start of that fermentation process, that also gets propagated up. So if you've got anything there at all, it's quite tricky to control it. So all of these steps, everything's autoclaved, everything's very, very clean. The biggest danger is contamination within the fermenter, not necessarily contamination within the drying process, because in the drying process, there's no growth taking place, right? So there's no opportunity to then propagate that contaminant. If you get contamination in the fermenter, especially if it's the start, it's a no-no. The batch is probably done. So it's quite an involved process, tricky to yeast. This is what some things look like. So on the top left, that's a rotary vacuum filter. And it's hard to see, but the middle picture is the yeast being cut away from the drum of the vacuum filter. And then on the right, you've got the vermicelli, the yeast kind of spaghetti that's being extruded through a plate. And then that is conveyed into the dryers. There isn't a picture of the dryer because it's just a big tank. It doesn't look very exciting. This is kind of how we do it. This is the kind of the propagation process. You start off with the level of sugar, because you have to start with some of the molasses in there because it makes up a large volume of the tank, right? So you start that fermentation process, the ethanol starts to rise and the yeast kind of slowly starts to grow. But as that sugar is depleted, you can then force the yeast to start to grow aerobically and respire. And you do fed batch, you start to feed molasses, inject it into the tank to ensure that there's something there. And the oxygen is at such a level that it creates biomass but doesn't produce as much ethanol. So that's the really tricky bit behind dried yeast production. As I say, in a brewery environment, a big brewery, they might do something like this, but most breweries will tend to be more of an anaerobic process, so you won't get as much biomass production when you're doing it. Then we have QC. So there's loads and loads of steps. And we're always trying to do this better. So our continuous improvement, we're always coming up with new methods to try and find things, new methods to look at SDA contaminants, that sort of thing. So we're always kind of trying to advance our knowledge in that area. But there's some key ones in there. So number 1 is the strain identity DNA test. So that's where you make sure that the Lallemand yeast culture collection sent you the right slant and you haven't just produced some baking yeast. So you have to ensure it's genetically the correct strain, obviously. And we would pick that up very quickly, if it was the wrong one, and somebody would get very, very badly told off. So that's never really been a problem. Viability, we would look at viability. So if you had problems within the drying process or the propagation process, you might find that your viability was too low. So you look at viability, and we have very strict limits for our viability. There's a limit below that we wouldn't release. Then we might look at non-Saccharomyces wild yeast, and that's actually a microbiology technique. So that's lysine. So a lot of your non-Saccharomyces wild yeast will grow on lysine, which is an amino acid, but many brewing yeast will not. Then we have our diastaticus test, so looking for diastaticus yeasts, and we've increased the level of sensitivity that we look at now. So we can detect I think it's like 10 to the 6, maybe we can detect like 10 to the 6 in 10 to 6, I can't remember. It's very low anyway. We're getting lower and lower and lower every time we try it. And it's something that we do a lot on. And then we'd look at diastatic activity as well. So we'd look at pressure buildup in flasks. We might look at gas production. You can look at starch plates as well. None of those are like, none of them are perfect. So you're always kind of comparing the genetic tests and the microbiology tests to come up with an answer, because there's always sort of downsides to everything. You know, there's no right answer. Fermentation kinetics, that's how fast the yeast is. So you know, we'll look at the CO2 production over a kind of trial fermentation in EBC tubes, which are big tall glass tubes that mimic fermentation sort of aspect ratios, and we just make sure that they're fermenting at the right speed. We'll look at total attenuation. So all of our tests are done on a standard malt extract in the lab. So you would always see the same level of attenuation. And if you didn't see the same level, you might indicate a problem, whether that be with yeast viability or some sort of contaminant. We'd look at bacteria, and we do lots of different tests for bacteria. Bacteria is actually usually not too much of an issue. We also grow, when we do our fermentations, we use hop acids as a biocontrol in the fermenter. So hop acids are active against gram positive, I think. Could be wrong, Now might correct me here. But yeah, they act as biocontrol in the fermenter because they are active against certain bacteria. Look at coliforms, which are specific to kind of wastewater contamination. We look at the vitality of the yeast, the color, the dry weight. So that does actually sometimes vary, the dry weight. So you might have different densities of yeast depending on how it dried. We do the shelf life, activity. We do sensory on everything, so every batch gets sensory. Lactic acid analysis, bottle conditioning tests for the specific bottle conditioning yeasts. So you have all these tests. And then after packaging, you do them all again. So you do the bulk yeast, and then you repackage it into 500 gram, and then you do it all again on the 500 gram. So that's the reason it takes so long. So it's basically a double process. So that's for us, one of the major costs of producing dried yeast is actually the QC process, because it's a lot of time in the lab to get it right. Oh, that's the end. So time for any questions.
There is a question in the chat. Roughly how much packaged dry yeast comes from one of those 1,000 hectoliter tanks?
So it does depend on the batch size and the strain. So if we were brewing something like Nottingham, which grows very efficiently in the tanks and produces a lot of biomass, you could get up to about 10 tons. If you're looking at a strain that's a bit more wimpy, for instance, we had a coal strain that we recently discontinued because it was hard to grow. You might get 1.6 to 2 tons off that. So it really does depend. One of the reasons for that is some strains want to go for Crabtree effect more than others. So the coal strain would prefer to ferment than it would to respire, and therefore, when you're growing it in the fermentation tanks, you get less biomass production, and then you get cell death during the drying process as well. So strains like that are very, very hard to grow. One of the reasons we discontinued it is because, one, the sales were low, but two, we'd planned to discontinue it, and then it failed like 3 times in a row, so it kind of discontinued itself. So yeah, it could be quite hard to produce strains like that.
Any questions? Yeah, Mirko?
Yes. Now I'm a big fan of the Nova Lager strain, which is why my final thesis for this project will be about the Nova Lager. It will include a test where I fermented the same wort basically at 10 degrees, which is the lowest limit you give as a supplier, and at 20 degrees, and then have people trying it. But I understood right that you do not see the need of a diacetyl rust.
Usually not, although I would always base it on sensory, so make check. But it should be quite low on diacetyl production. It still produce some, so if it peaks and if it's at flavor threshold and it isn't given a chance to reduce, you could still get some, but it produces way less. So it's more like produces less, but it does still produce some.
Mm-hmm.
But yeah, I best do it on sensory. So if you taste it and it's fine, go to the next stage. You don't need to wait any further.
All right. Probably in the course of writing a thesis, I will have a bunch of questions. Can we reach you somehow?
Yes, of course. Yeah, just email me. Put my email address in the chat, and then you have it.
Perfect. Thank you.
And I also have it if the people who listen afterwards, they can't see the chat. So if you be in touch with me, I can give them.
Mirko, you should write it in English so you can send it.
I will probably anyway, because
good,
honestly, it
yeah.
makes my life much easier.
Cool.
Otherwise, I'll have to ask ChatGPT if they can speak Swedish. It probably can.
There is.
That's how I go live in Sweden with ChatGPT.
Great. Any more questions? No. No, that was wonderful, and thank you so much.
No problem at all. Yeah, it's fun. Enjoy doing it.
Yeah, it's good, so good to hear from you, and that you are willing to do this for us. It's amazing.
Yeah,
So,
yeah, anytime. So if if you want us to do it again, we're more than happy. And I don't know if you ever do anything physical, but I do come to Stockholm sometimes, so I can.
yeah. Okay, great. That's good to know. Thank you. So we end it for today, and I want to say thank you again. And yeah, thank you.
Yeah, no
Thanks very
problem at all.
much.
Yeah, no problem at all. And I'll hopefully see you guys around somewhere at the next thing. I'm not going to be at the Nordic Brewers Conference in Copenhagen in a few weeks, but I can't make it. So I've got a baby coming in like 2 weeks. So
Great.
so I'm not allowed.
Great.
Cool.
Never mind. Okay, well, thank you very much, and
Bye.
yeah, have a great rest of your day.